So good morning, everyone. I cordially welcome you all to the today's International E-Conference on Microbial Pathogenesis and Recent Advances in Diagnosis of Omicron, organized by the Department of Microbiology, Palamuru University, Mahabubnagar, Telangana, in collaboration with Indian Women Scientists Association, Microbiologist Society India Association, and IQAC, Palamuru University. On the behalf of Microbiology Department and Palamuru University, I, Dr. Shantipriya Ajmera, the convener and organizing secretary of this event, feels a privilege to welcome you all to the inaugural session of today's conference. I would like to start inaugural session by inviting distinguished dignitaries, guests of honor, and invited speakers. It's my great privilege to invite the chief patron of today's conference, Professor L.B. Lakshmikant Rathod Garu, Vice Chancellor of Palamuru University. Hearty welcome to you, sir. It's an immense pleasure to invite the patron and chairman of today's conference, Professor Pindi Pavan Kumar Garu, Registrar, Palamuru University. Please welcome, sir. A warm word of welcome to Dr. Noorjahan ma'am, Principal and Chairperson of this conference. Please welcome, ma'am. It's my great honor to welcome Professor Arvind Deshmukh, sir, National President of MSI. We are very happy, sir, with your collaboration. Thank you, sir. It's a glad welcome to Dr. K. Ratna, ma'am, convener of the Indian Women Scientists Association, Hyderabad chapter. Please welcome, ma'am. And we are very happy with your collaboration, ma'am. Thank you. It's an immense pleasure to invite Professor Anjali Devi, ma'am, former professor and HOD of Food and Nutrition, Usmania University, and former registrar of Sri Padmavati Mahila University, Tirupati, and secretary of Indian Women Scientists Association, Hyderabad chapter, and today's keynote addressee. Please welcome, ma'am. I truly desire to invite the eminent speakers of this conference, Professor Sharmisha Banerjee, ma'am, from Central University, Hyderabad. Please welcome, ma'am. It's my pleasure to welcome Professor Sai Gopal, sir, Vice Chancellor of Cluster University, Kurnool, and Professor of Virology Department, Tirupati, SV University. Please welcome, sir. It's a glad welcome to invite Dr. Gokul Sebasan, Associate Professor in Manipal Medical College of Malaysia. Please welcome, sir. It's a gracious welcome to Dr. Ruxana Rayana, ma'am, Associate Professor in the Department of Microbiology, US Bangla Medical College, Bangladesh. A warm welcome to you, ma'am. Dear participants, I am very glad to extend a warm welcome to you all. My heartfelt gratitude to all of you. Now I request you all, please join with us in lighting the lamp.
Output line. Madam, unmute. Madam, unmute. Shanti Priya, madam, unmute చేసుకోండి. You are muted.
I am very happy to speak about this conference. It is a big event for us. The program is shaped in a way to attract your interest in the present scenario. We hope it would be a successful event. From 2020 till today, we are experiencing the effect of COVID-19 to different variants like alpha, beta, gamma, delta to Omicron. During this period, among us lost our loved ones and we missed our near and dear. As we know, microorganisms, both beneficial and harmful, microbes themselves, they evolved specific tools to enter into the host, and consequently, they can damage the host tissue and spread over the body's organs, and they develop the disease or infection. So the severity of the infection may vary in the individual based on pathogen virulence factor and host defense system. So the main aim of the conference is to create awareness among the participants about microbial pathogenesis, its adaptations to the environment, the progress of the current outbreak COVID-19 pandemic to different forms, variants, and associated mycotic infections, its diagnosis and administration of vaccines to control the pandemic. At this conference, we have eminent speakers. They will discuss and deliver their talk related to these themes. I am truly thankful to the speakers for their humble response towards us and for accepting our invitation. I appreciate all the participants for attending this event. I hope all the participants will be benefited from this conference. Thank you, one and all. Over to Mercy.
Thank you, Dr. Shanti Priya, ma'am. Good morning to all the respected dignitaries and all the attendees of this webinar. It's a moment of extreme pleasure for me to welcome our dignitaries to give us their suggestions and encourage us through their valuable words. First, I would like to invite our chairperson, Dr. M. Noorjahan, ma'am, Principal of PG College, Palamuru University, to give away her valuable speech. Over to you, Noorjahan, ma'am.
You go forward, Mercy.
Next, I would like to invite our patron and chairman of the conference, Professor P. Pavankumar sir, Registrar of Palamuru University, for the inaugural speech. Over to you, sir.
Yeah, thank you, ma. Is it audible?
Sir, you're audible.
Yes, sir.
So, greetings from Palamuru University. A warm welcome to all the dignitaries, eminent speakers, academicians, scholars, and student friends, and my dear media friends. For the 2-day International E-Conference on Microbial Pathogens and Recent Advances in Diagnosis of Omicron, organized by Department of Microbiology, Palamur University, Mahbubnagar. First of all, I would like to thank all the eminent speakers for their acceptance to participate in this e-conference, and they are ready to share their invaluable knowledge to our audience. I really appreciate Dr. Azmira Shantipriya, who has taken initiation to conduct this e-conference, and with the collaboration of Indian Women Scientists Association and Microbiology Society of India, along with the Internal Quality Assurance Cell, Palamur University. So even I would like to thank Professor Aravind Deshmukh, who is a person who is a president of MSI, who is behind this program. In the past 2 decades, the world has faced several infectious disease outbreaks like Ebola, influenza, SARS, MERS, Zika virus, had a massive global impact in terms of public health and economic disruptions. Most burning and relevant topic has been taken for this conference. And most importantly, whenever we are into new advanced technology, and we have seen that nature creates some challenges. So, and it has been demonstrated numerous times, like plague, a Spanish flu, yellow fever, polio, AIDS, Ebola, Zika, and other bird flu, swine flu, and COVID. Now we are seeing that Omicron. So as all now, this a diverse group of microbes are essential for the sustainable biosphere. Generally, in general, microbes are more important and crucial in nature, and they are playing very important vital role in the nature. And microbes are key to human survival and a huge reservoir for resources for human welfare. But at the same time, what we have seen that the other part of the coin, so some of the pathogens, we are facing problems with the pathogenic organisms, and we should be ready with these challenges. And like coronavirus, a large virus family that causes illness ranging from common cold to acute respiratory infection. The infectious severity may manifest as pneumonia and acute respiratory syndrome and even death. However, since SARS and MERS outbreaks, the virus have been studied in greater depth, which has accelerated vaccine research. RNA viruses, which have remarkable adaptability and multiply to overcome any obstacle that is imposed on them. This is due to their extremely high replication rate and proclivity for mutations. As a result, its high mutation rate can be regarded as a greater barrier to COVID treatment. Another reason is that most RNA viruses lack mechanism for detecting the and repairing mechanism, resulting in polymerase chain reaction, or you can say polymerase errors. Because of the error-prone nature of polymerase activity and the lack of proofreading mechanism, RNA virus genome requires mutations. This RNA virus genome, acquire mutations and exist as forms of genetic variants like alpha, beta, and delta, and other Omicron variants. Vaccination has played a key role in immunization. The recent emergence of Omicron variant further emphasizes the importance of vaccination and boosters. Treatment of some monoclonal antibodies may not be effective against these infections. The coronavirus genome consists of 30,000 nucleotides, and it encodes 4 structural proteins which are important, and nucleocapsid protein like membrane protein, spike protein, and envelope protein, and other several non-structural proteins. The N protein codes the viral RNA genome, which plays a key role in the replication and transcription. For diagnosis purpose, we are using RT-PCR, which is nothing but a real-time reverse transcriptase polymerase chain reaction based on the CT values, like a cycle threshold number for cycles required to amplify the viral RNA to make it detectable. The CT values determines the viral load, which is less than 35 cycles or more than 35 cycles. On the other hand, the rapid antigen test is also used for the antibodies against a COVID. So far, chloroquine and hydroxychloroquine, remdesivir, these medicines are extensively used for the treatment of COVID. But now, definitely, we should look forward for that 2-deoxy-2-glucose, the 2-DG, which has developed by our DRDO India. And now recently, what we came to know that Wuhan Academy of Sciences mentioned that even the new variant, the neo-COVID, it is just a step ahead of one more nucleotide mutant. So it may be arising, and it is uncurable, and where we can see the solution to face such problems. So time to time, the worldwide scientists has to sit together and to have look into this whole genome sequence of these variants and how these variants are changing time to time and affecting on the human sustainability. So here we have learned enough lessons from last 2 years. So now it's time to take care of every human being life, and we should more careful towards our food habitats and boost up our immunity and resistance power. So to face upcoming challenges, even we should maintain our personal hygiene, our good habitats, and also we should have some good practices like yoga and meditation and natural physical exercises, which will definitely help to boost up our immunity and resistance power to face in near future the challenges which are caused by the viruses. I really appreciate once again Dr. Shantipriya Madam and her team for conducting such nice, excellent program. And I also really thank all the speakers and the participants. So definitely you will be again enlightened with the new knowledge. Thank you once again. Thank you one and all. Thank you so much.
Thank you, sir, for giving a detailed talk about coronavirus and various outbreaks. Thank you very much, sir. Next, I would like to invite our chief guest, chief patron of the conference, Professor L.B. Lakshmikant Rathod, sir, Vice Chancellor of Palamuri University, to give away his valuable speech.
Hello.
Over to you, sir.
I'm audible?
Yes, sir, you're audible.
Very good morning to you all. Respected Registrar of Palamur University, Professor Pawan Kumar Garu, YST2 Vice Chancellor, Palamur University, Dr. Masudan Reddy Garu, and the keynote speakers today's Professor Shastri Banerjee and Dr. Gokul Shankar, and today's organizing secretary of this two-day international conference, Shantipriya, Professor Shantipriya Garu, and all the department of microbiology, and my dear young friends, those who are participating in the two-day international seminar. It is my eminent pleasure to speak as the chief guest of this two-day international e-conference on microbial pathogenesis and recent advances in diagnosis of Omicron being organized by the Department of Microbiology, Palamur University. The current 21st century is witnessing emergency of many infections, diseases which are caused by various microorganisms. The relationship between a microorganism and a disease is not a new concept, as it was established before hundreds of years. But it is ironic to know that till today we are unable to detect the role of microorganism in certain infections, as scientists fail to isolate the cavities agents. The diagnosis and treatment of such influences are always posing a challenge to this community. We all know diagnosis play an important role in finding out a remedy for any diseases. An economic, simple, and less time taking diagnosis process is always help us to control the spread of any infections. This we have witnessed with the COVID-19 pandemic diseases, where it has challenged and humankind with its alpha, beta, gamma, delta, and Omicron variants, and we do not know what would be the next to come and how it will impact globally. The vast researcher and many conferences and seminars need to be conducted to share the information globally from entire world. I deeply commend the initiative taken by our microbiology department, and I am congratulating the convener of this two-day seminar, Dr. Shantipriya, and her team in planning this two-day international e-conference, which would cover very interesting topic on the above subject. And I am very much thankful tomorrow's keynote speaker, Professor PVR Sai Gopal, Vice Chancellor, Cluster University of Kurnool. He will deliver tomorrow's lecture. And I hope the session are going to be informative and discussing that are going to be held would be meaningful and fruitful for 2 days. I wish the International E-Conference all success. Thank you, one and all. Thank you.
Thank you, sir.
Thank you for your valuable words, sir.
Thank you.
Next, I invite our guest of honor, Dr. Anjali Devi, ma'am, former professor, HOD of Food and Nutrition, Usmania University, former registrar of Sri Padmavati Mahila University, Tirupati. To introduce the speaker, I would like to invite Dr. Kishore sir, HOD, Department of Microbiology, Palamuri University. Over to you, sir.
Good morning, one and all. It is my privilege to introduce Professor Anjali Devi, the keynote speaker of today's conference. Professor C. Anjali Devi served as professor and head department of Food and Nutrition, Usmania University, Hyderabad. She was former registrar of Sri Padmavati Mahila Vishwavidyalaya, Tirupati, and present secretary of Indian Women Scientists Association, Hyderabad. To her credit, she published around 40 national and international research papers, 10 books, and completed 30 major research projects funded by USAID, UNICEF, German Social Service Society, ICRISAT, Ford Foundation, ICAR, UGC, etc. She developed a web portal on nutrition, that is nutritiondatabank.com. She guided several students for their PhDs and presented several papers at international conferences abroad, like Paris, Dallas, Minnesota, Arizona, Dubai, Thailand, Indonesia, Amsterdam, etc. She bagged several awards in her career, like Scientist of Eminence Award for outstanding contribution in food and nutrition science. Special commendation certificate, recognition certificate was given at international conference at Paris, France. She is also a member of several academic bodies, like EC member of Nutrition Society of India, Vice President of Indian Dietetics Association, Secretary of Indian Women Scientist Association, Vice President of Usmania University Teacher Association, Associate Member of AP Academy of Sciences, etc. Professor Anjali Devi was very active during the hard times of pandemic. She gave online dietary counseling to patients and introduced complementary therapies like medical, nutritional therapy, physical therapy, etc. for several ailments. She created a help desk for dietary tips to improve immunity and also publicized them through telecast in ETV Sukibava, ETV Live, Maha TV, etc. and also published several articles in local dailies. Also, Professor Anjali Devi was actively engaged in maternal and child health projects in remote villages of Telangana, which is now taken up by the government of India as ICDS program. She is an active member in several other activities like nutrition information technology services, annual health camps, yogas, tailoring, Carnatic music, etc. Her CV is very broad. I have made it cut short. With this brief introduction, I now invite Professor Anjali Devi to enlighten us with her lecture. Over to Professor Anjali Devi.
Thank you, sir. You have given a very detailed description about myself. Thank you very much.
Madam, before that, very good morning to you, madam. I welcome you, madam.
Thank you, sir. Thank you very much. It's very nice to hear from you, sir. Thank you very much.
Thank you, madam. Thank you.
Thank you.
Okay, carry on, madam.
Professor L.B. Lakshmikant Radhod, Vice Chancellor, Palamur University. Professor Pavan Kumar, Registrar. Dr. Nurjahan, Principal. Dr. Shanti Priya, Staff Department of Microbiology. Speakers from various countries, invitees, and research scholars and students. It's my privilege to you, sir, and to Dr. Shanti Priya for inviting me to address the August gathering today. I start with saying that there are good bacteria which are present inside every human body. They are present everywhere. Therefore, microbes have both beneficial as well as deleterious effects on safety and also on quality of foods, thereby a concern to today's major public health problem. I would briefly touch upon the microbes which are related to nutrition and health. The majority, I think we can divide into three major categories. That is microbes involved in the manufacture of foods, in food storage, and in transmission of diseases. So with regard to the variety of foods that are manufactured using the microbes, at the household level, we have bread, which is made porous due to the fermentation process through baker's yeast. We have fermentation of dough to make our daily breakfast in the food, that is idlis and dosas. Kimchi, sauerkraut, yogurt, cheese are produced by bacterial fermentation. Toddy, a traditional drink of South India, which is made by fermenting sap from palm trees. Fish, soya bean, bamboo shoots are fermented to produce special food. Swiss cheese and Roquefort cheese are due to fermentation. In the industrial category, we have wine, beer, whiskey, brandy, or rum. Production of antibiotics like penicillium to kill or retard the disease-causing microbes. The brewer's yeast is used to produce ethanol. Yeast ferments some of the cereals and fruit juices. We have Aspergillus niger, which is used to produce citric acid, and we have the Acetobacteria aceti producing acetic acid, Lactobacilli producing lactic acid. Streptococcus produces streptokinase used as a clot blaster in the medical practice. In the sewage treatment, we have in the sewage treatment plants with microbes to make it less polluting, and the excreta of cattle is used as a biogas. In the natural environment, microorganisms have very specific jobs. They are responsible for recycling the nutrients in the soil and also purifying the water. So there are certain microorganisms which cause spoilage of foods. These are very important. In fact, there is the scientist Los Lorenzo reported that microbiology is important to food safety, production, processing, beverage, and at every stage of storage. We have our traditional techniques like salting, dehydration, and other processes which were used from ancient days in the preservation of these foods. But now we have the modern technologies like pasteurization, sterilization, good hygiene, and good manufacturing processes which delay spoilage. But still, food is exposed and remains susceptible to spoilage and contamination with the presence of pathogenic microorganisms throughout the food chain, retail shops, restaurants, and even at the home of consumers. Nutrition and infectious diseases relate to each other. First, nutrition affects the development of human body immune system, food poisoning, intestinal diseases, and infectious diseases. Thus, there is a direct link between microbiology, nutrition, and health. So microbial diseases are transmitted through 4 major routes. One is through the respiratory tract, which we are now facing. The entire family of corona, starting from gamma to the Omicron, all are passed through the respiratory route. That is through mouth and nose. An example is the flu that gets originally is through the transmission through the respiratory tract. Now we have the next is the gastrointestinal tract, that is through the mouth and oral cavity. An example is cholera. Urogenital tract caused by E. coli, which is causes cystitis, and breaks in the skin causes tetanus. So now the impact, let us just briefly discuss on the impact of microbes on health. To start with, the development of human immune system begins from embryonic stage. If the pregnant mother receives a good balanced diet in the first trimester of life, definitely the baby is born with a normal weight, and throughout the life span, the baby till the old age or to whatever it is, the boy or the girl retains the immunity and is very and is not affected by any type of environmental pathogens. But if this phase is neglected, then the gap will continue all throughout the life. Breastfeeding is the second most important system to develop immune system. This led to importance of feeding practices in the first 1,000 days of life, and now we also concentrate on the first 1,000 days of life. So a scientist from London School of Hygiene and Tropical Medicine stated that a malnourished child who did not get enough proteins, vitamins, and minerals is prone to infectious diseases and does not respond well even to the vaccines. So now we see quite a number within any family. If a vaccine is, even after administering the vaccine, the person gets the disease, which was mainly because of the natural immunity that the person did not have throughout their life. Immunity is something which you can't get overnight through overnight eating of proteins and or taking a lot of concentrated vitamins, but it has to be developed over a period of time. So we need to take care of all our children from now onwards in all different categories to see that they grow up with a good, strong immunity system. The malnutrition infection cycle shows that inadequate dietary intake leads to weight loss, growth faltering, lowered immunity, mucosal damage, disease incidence, appetite loss, nutrient loss, malabsorption, altered metabolism, which are again based on weight loss, the various types, and the cycle gets repeated. That vicious cycle once again gets repeated. So microbial contamination transmitted through water and food can cause severe diarrhea and infections in children. Waterborne diseases are due to using unpasteurized milk or undercooked meat. And consumption of contaminated water and food leads to amoebiasis and several other diseases like respiratory infections, hepatitis, tonsillitis, rheumatoid arthritis. And studies conducted in several countries like Kenya, Bangladesh, Colombia reported that there is a reduction in blood vitamin A, riboflavin, vitamin C, iron, zinc, protein levels due to infection. In short, it can be concluded that microbiology has a close relationship with nutrition and health, and they are all very closely interrelated. Thank you one and all for your patient hearing once again. Over to.
Thank you, ma'am.
Thank you, madam.
Thank you for the detailed talk about various pathogenic and non-pathogenic microorganisms, and also throwing a light on nutrition and immunity. Thank you very much, ma'am. Next, we will enter into the technical session of the webinar, for which I would like to invite our first speaker of the day.
Madam, appreciation slide.
Sorry. Before that, we would like to.
Madam, please accept our appreciation certificate. Anjali Devi, ma'am, please receive our appreciation certificate. Thank you.
Thank you very much.
Thank you, ma'am. Next, I would like to invite first speaker of our day, Professor Shikimate Banerjee, ma'am, Department of Biochemistry, School of Sciences from Central University of Hyderabad. To introduce the speaker, I would like to invite Dr. Shantipriya Ajmera, ma'am. Over to you, ma'am.
Thank you, Mercy. I feel it is a great privilege to introduce about Professor Shikimate Banerjee, ma'am. Ma'am did her graduation and post-graduation in zoology from Ranchi University, followed by a doctoral degree from University of Hyderabad in the year of 2005. She did her doctoral research at Center for DNA Fingerprinting and Diagnostics, Hyderabad, on tuberculosis bacteria, Mycobacterium tuberculosis. She joined as assistant professor in the Department of Biochemistry of University of Hyderabad after her postdoctoral studies at Lerner Research Center, Cleveland, USA. She is the recipient of several national awards, such as INSA Young Scientist Award in the year of 2005, BioAsia Innovation Award 2008, Innovative Young Biotechnologist Award 2011, and National Women Bioscientist Award 2006, recipient of the Chancellor's Award 2017. She is presently a professor in the Department of Biochemistry, School of Life Sciences, University of Hyderabad, and teaches immunology and enzymology. As an infection biologist and immunologist, her research focus is molecular pathogenesis and immunology of mycobacteria, HIV, and mycobacteria HIV comorbidities, the laboratory has added important baseline information on molecular mechanisms behind the pathogenesis of Mycobacteria and HIV during both mono and co-infection, deciphering new molecular mechanisms, biochemical pathways, potential drug targets, and biomarkers. Madam was published more than 50 research articles, 4 book chapters, and 2 applied patents. Ma'am, I am really very thankful to you, and please, I welcome to you, ma'am.
Thank you so much. Am I audible?
Yes, ma'am. Are you?
Very new platform, but it is such a pleasure to be with all of you. So the last interaction I had is a couple of years ago, and it still is so fresh in mind that I have always enjoyed coming back to your university. Thank you. I thank the organizer for giving me this platform, although I think Omicron is the one which is so much in news. But then I decided that there is something which we should talk about is what I am expert in, and also I feel that something which we require to talk about is tuberculosis. At the beginning, I once again thank you for having me here, and if you permit me, I will share my slides and talk about the molecular pathogenesis of Mycobacterium tuberculosis, touch upon a bit of the research that we do, and try to take, because I was also told that there will be a lot of research scholars and master students in the audience, try to take them that in this era where we are talking about infectious diseases, how important it is to understand the molecular mechanisms, because that is what eventually leads or help us in designing various strategies to control them. We have seen that in the last 2 years, how important it is to have a very interdisciplinary scientific approach to control many of the infectious diseases. And that is the primary essence of my talk. And so I'll start sharing my slides now. If that may I, if the organizers permit, may I?
Yes, ma'am. Yeah, continue, ma'am.
Yeah,
Yes.
give me a time. If because this is a platform which sometimes there can be connectivity issues. Somebody should prompt me if I lose my connection, and I should not be talking to myself, because I'm not going to see the audience otherwise. And I'm sure all of us have faced this while online teaching all the time. So please prompt me, and if it becomes really, the connectivity becomes an issue, then I may have to switch off my video. So, is it visible?
Yes, ma'am. Yes. It's visible, ma'am.
And it is also visible in the slide mode, right? With PowerPoint
Yes.
mode. Right.
Yes, ma'am. Yes, ma'am.
Okay, thank you. So today I would want to talk on, I would be talking on molecular pathogenesis of Mycobacterium tuberculosis. And I'm sure the audience, almost all of us have heard about the disease tuberculosis, which is a highly contagious and is commonly caused as TB. The disease is generally known as TB amongst common man in the society. The lab, as I was mentioned, we work on 2 pathogens, Mycobacterium tuberculosis, the TB-causing bacteria, and human immunodeficiency virus, HIV. And we try to study their molecular mechanisms, how are they causing, infection, how they reside inside our cells, and how they try to propagate. What does the host do to control these infections? And this one we study both at the level of individual pathogen as well as when there is a co-infection, because there are a huge number of infections of Mycobacterium. All the 3 aspects with our main goal is to have a translational approach where we can identify biomarkers for early detection. I believe it was also spoken a few minutes back how important it is to have an infection detected early so that the therapy can be started early as well. At the same time, we have understood that how it is important to identify new drug targets and design small molecules to have intervention strategies ready for those pathogens which are going to be developed drug resistance at some point of time. We also do a lot of basic biology in understanding these pathogens. But today I will be primarily concentrating myself on this. I'll just. So today I will be primarily talking about Mycobacterium tuberculosis. Initially, I will be just concentrating on trying to tell that why this tuberculosis is such a problem, and especially in the context of India. And what is it today, especially in the COVID-19 pandemic area, or how alarming it is in the COVID-19 pandemic area, and then take the audience to understand that how the molecular mechanisms, when we understand about these, helps us in designing strategies, intervention strategies, and disease challenges to address the disease challenges, and possibly end with a note that we do require a lot of interdisciplinary research to address many of the infectious diseases that are either emerging or are persisting in our community and society. So tuberculosis, as we know, is a very common, highly contagious and aerosolic infectious disease, which is caused by bacteria, Mycobacterium tuberculosis. Although Mycobacterium tuberculosis is the primary bacteria, mycobacteria that we know causes the TB disease, but there are a number of other species that can also infect human beings and can give a kind of symptoms that we have that of tuberculosis. In the lab, when you grow them, it looks like this, buff kind of colony, and this is otherwise it's a long, non-motile, it's a rod-shaped bacterium. The bacteria, as we know, gets its name from a very thick, fatty, layer of cell wall of mycolic acid, and that actually also is responsible for a lot of its virulence property, including sometimes to avoid the antibiotics that are given to it. Is a very slow growing bacteria, and most of MTB, although is a very active and a very highly infectious pathogen, but many of them, they can infect mankind in very opportunistic way. When we say opportunistic pathogens, Professor Anjali Devi just now talked about how important it is to have a very robust immune system. So good thing about our immune system is most of the time it is very capable of taking care of many of the infections that possibly we inhale every day, and we don't even know, because most of the time the immune system, a robust immune system, takes care of it. But whenever there is there is a compromise in the immune system, immune health, and that can be for a number of reasons, including malnutrition, including some kind of other comorbidities that we talk about, some other kind of diseases that if a person may have, some other physiological disorder, or sometimes even stress can compromise our immune system. And at that point of time, such pathogens like as listed here can become opportunistic in nature, and the infection can manifest in the form of a disease. So tuberculosis, the primary site of infection definitely is lungs, but there are also sites at which the Mycobacterium tuberculosis can cause infection, and we call them extrapulmonary TB. It can be in the pleura. It can be in the brain, which we call this as meningitis, meningeal TB, the lymph nodes. There can be spine TB, there can be bone TB, and such tuberculosis are very, very difficult to diagnose, because the symptoms are so diffuse that most of the time it escapes our recognition that there can be a possibility of a Mycobacterium tuberculosis infection. So extrapulmonary TB and detection of the extrapulmonary TB at an early stage to start a therapy remains a challenge even today. So if you do look at the mode of transmission, this is just to kind of brush up. We all know that the mode of transmission is again aerosolic. While I'm talking about these mode of transmission, the symptoms, one may want to also kind of correlate how similar it is to the one which we are seeing day to day during our COVID-19 pandemic. It is an aerosolic infection. It also spreads by droplet infection. It also is inhaled and is the route of entry is through the respiratory tract. And anybody who's in close contact are at the highest risk of being getting infected. The very hallmark of the TB disease symptoms are persistent cough. While we see that these days you do get this flu-like symptoms, sometimes there's a itching in the throat and there is a cough. But for TB, it is generally if you have a continuous cough, an unexplained cough, and there is no reason, and it doesn't the cough pursue. At the same time, there is a severe weight loss. That kind of definitely should alarm a person that one should go for a TB diagnosis. And at a later stage, cough becomes productive. There can be huge amount of mucus, and with the mucus comes blood. But by that time, it would mean that the disease has really is been there for a long time and has caused considerable damage to the lung. The TB generally, it is, evaluation of TB generally is done through physical examination, chest X-rays, where you see this characteristic hallmark of TB in the lung. You have granuloma, there are patches which can be seen. But these kind of patches, we have also been reported in case of this recent pandemic as well. But a clinician would be able to distinguish, and along with that, when it correlates with the clinical symptoms, there is a tuberculin test, and then there are also bacteriological examinations that are done in the form of culture, and advanced tests like interferon gamma release assays are sometimes used as a supplementary test as well. So if you look at the status of tuberculosis, it is a huge problem globally. It's a very, very global problem, and it is the second leading infectious killer after COVID-19 now. So it was the leading, and SARS-CoV-2 has taken over it, but a huge amount of death is caused even to this day. And if you look at the color that India is being painted here in the world map, and it is not a very encouraging to read from the WHO report that 8 countries account for 2/3 of the total, with India leading the count. So India has a huge load of MTB infection, most of them in the form of a latent infection. And at any point of time when there is a compromised immune system, it can flare up into a disease. So we are almost, people sometimes say it is like a ticking bomb. So what does it mean for, the tuberculosis means in case of the situation that we have in COVID-19? The tuberculosis disease management requires a very close contact between the patient and the doctors. And so we have this news, which is a very recent news, and it is definitely not very encouraging. Tuberculosis deaths are in rise for the first time in more than a decade due to this COVID-19 pandemic, because it has really disrupted the healthcare that was extended to such mass number of tuberculosis patients. So much that this time's theme of the World TB Day on 24th March was that the clock is ticking, and it was flagged that 1 person dies of tuberculosis every 20 seconds, and that is a very, very alarming number. So but WHO has now decided and has put forward that it requires to end TB by 2030. And when it has released that how many of them are having tuberculosis in HIV background, TB alone background, and TB with resistance to the existing therapy that is called multiple drug resistant therapy. If you see these, all the squares, India appears in all the square, and that is not a very encouraging situation for us. So what are the problems associated with tuberculosis? If we try to understand that, first and the foremost thing is TB is curable. If TB is curable, then it is not acceptable in today's era that we have so many deaths and infection, and yet it remains a major health problem. So what are the reasons that we may want to look at? What are the area that has huge lacunae that we have to fill up in order to take care of this disease? Is one thing is that there is still we have a very failure of effective vaccine protection in adults. If most of you know that we in India, at least we get all of us vaccinated with BCG. And although it gives fairly a good protection, possibly up to for 10 years and or 10 to 12 years of our age, but it has been shown that it is not a very good vaccine which gives protection in adults. So we require to have more effective vaccines which will generate long-term memory and gives us protection. One of the another challenge is the lack of early detection. When I was telling you about the symptoms of TB, and if you see that they're so diffused a symptom, if we have a little bit of cough, if we have a little bit of malaise, it can be confused with just any disease, and most of the time we kind of ignore, and and that if the bacterial load is not very high, it is not even very easily detected. It takes a long time for detection, and that delays the onset of the therapy itself, and that possibly is another challenge that we have in TB disease management. One major challenge is the development of drug resistance. To begin with, which I will deal with it a little bit later, there are 4 drugs that are used for TB therapy. But the TB therapy is a very long therapy. It at least is a 6-month therapy. As a result of which, many of the patients, they discontinue the medicine when they start feeling good. This non-compliance or very random and use of medicines adds led to the selection of drug resistance, and the first-line drug-resistant strains have developed into multi-drug resistant strains, and now we have strains which are resistant to the second-line drugs as well, which are called extreme drug resistance tuberculosis. And we are seriously very worried that what will happen if there are strains which are circulating in the community which are resistant to all the drugs that are available for treatment, and there is complete drug resistance to it. So this is one of the major challenge because of which we require to really look into the pathogenesis of the TB and come up with some new strategies to regulate it. Tuberculosis, like as we talked about COVID these days, or SARS-CoV-2 infection, has more severity during comorbid conditions or in a co-infection condition, and shows a huge synergistic effect with comorbidities like dyslipidemia, diabetes, or HIV background. So there is a increased pathogenicity, the more severe pathogenicity of tuberculosis, TB, when you have a comorbid another patient, the person who is suffering from another kind of disease. So this kind of tells us how important it is for us to address this at the level of our society, and especially there's a high burden of TB in India, and we require to really make sure that we participate. And our government's long-term goal also is to eradicate tuberculosis from the society. So having understood that what the problem tuberculosis is, then let me very briefly tell you, before I take it to the research part, that how our immune system responds to MTB. We these days most of us are very aware of a lot of immunological terms, and thanks to SARS-CoV-2, the terms immunity, vaccination, the importance of vaccination have become a household name, and that's a very, very good thing that awareness has spread throughout the society. So Mycobacterium tuberculosis host-to-host transmission is also through aerosolic infection. It is inhaled through in the droplets when a person is sneezing or a person is coughing, and it reaches another person. And in the aerosol, in our when it is inhaled inside, there are your airway epithelial cells, which are there. They are the first to kind of recognize that there is a bacteria that has entered, and they recognize it through their pattern recognition receptors and try to generate an environment which will be have a certain anti-bacterial properties through antimicrobial peptides or other, there is mucus secretion, so that it gets trapped there and it can be, start discouraging the complete entry into the lungs. So inside the lung, when it enters, it is the alveolar macrophages that are the first site where the MTB is engulfed. And when the MTB is engulfed, this alveolar macrophages are the sites where this pathogen also lives. So if the macrophage has an upper hand and the load of the bacteria is not very high, then a number of actions are taken within the macrophage, which we call as innate immune response. This innate immune response, basically tries to generate a lot of reactive oxygen species. It is engulfed in the phagosomes, and the phagosomes are highly, they're activated, they kill the bacteria, and there is a very active secretion of a number of pro-inflammatory cytokines. The pro-inflammatory cytokines also, the local microenvironment is changed in such a way that it encourages presentation of the antigens, bacterial antigens on the surface of this antigen presenting cells, so that the antigen presenting cell, when it goes to the nearest lymph node, can activate a number of effector cells, especially the T cells. So what happens in another some 7 to around 8 weeks time, there is a very active cell immunity that develops. I'm not going to go into the details of the immune response here, but the adaptive immune responses come into place, effector T cells come into place, pro-inflammatory cytokines and a lot of memory T cells are also generated during that time. Now, a hallmark of tuberculosis infection is the generation of a granuloma. Okay. Now, the granulomas are a very specialized immune structure, which is actually formed by an interplay of both the host and the pathogenic factors. These are a set of immune cells, which includes activated macrophages, monocytes, dendritic cells, and also are encased by a lot of activated lymphocytes. And above that, there is also can be fibroblast cells, giving them a case-like structure inside which the mycobacteria are trapped and contained, and they are not permitted to disseminate anymore. So, and either they are there, therefore they are either killed or they remain latent there for ages. There can be very, very long latency period. So what we also understand about, and not to get worried about this very complex picture here, is that cell-mediated immunity by a number of T cells, which release IFN gamma, which further activates this macrophages, is an important event in the containing the macrophages, containing the mycobacteria, and allowing the macrophages to actively kill them and clear them from the system. So pro-inflammatory cytokines play an important role, and the activation of various subsets of T cells also play a very active role. So TNF-alpha, IFN gamma, these are the critical cytokines that are important for containment and clearance of this particular pathogen. Okay. So having known this, what is very, very clear is that that most of the time, when suppose there is an exposure, if there are 100% 100 people who are exposed, 70% people can clear it. And in some of the people that the infection can prevail. Amongst the infected people, even if there is an infection, the disease manifestation into an active TB is again for a very limited people. And that active TB is a good news that it is curable with a very long therapy of around 6 months, but some percentage of it will still results into death. In the event, if it is not an active TB, much of it is hidden in the body, and it is hidden in the bodies because immune system is able to control it and not allow it to propagate. And at the same time, pathogens also remain completely quiescent, doesn't propagate, and tries to evade the immune system, tries to give immune system, it hides from the immune system and is not recognized for the immune system to be cleared and remains latent. And now what we really do not know, that once this immune system becomes compromised, this latent TB can become active TB, and that can be because of a secondary infection. It can be even SARS-CoV-2 infection that can do that. It can be any other kind of comorbidity or any kind of other situation. If you have an accident and you are in hospital, your immune system is compromised. So what is good is that it is always a tug of war. Sometimes host wins, sometimes pathogen wins. So what is the take-home message that we get from here? The take-home message is that we do have the capacity, that our body has the capacity to fight this pathogen. Sometimes if it wins, we require to recognize those factors which allows it to win, whether it is a bacterial factor or whether it's a host factor. If we know what allows the pathogen to win, we may be able to regulate that. We may be able to design a therapy or a strategy to control the tuberculosis pathogen from winning. And once we can stop it from winning, the immune system has the capacity to clear it. So the present drug therapy, as I said, is a 4-drug therapy, which is from isoniazid, rifampicin, parazinamide, and ethambutol. Mostly they target very critical points. They target cell wall synthesis and RNA polymerase, as well as there are protein synthesis are targeted. Okay, so what is if we have a therapy, what are the issues with this particular therapy? The issue is this a very, very long-term treatment, which leads to non-compliance. There are drug resistance that are coming to isoniazid and rifampicin. These are the first-line drug and very important drug, and we call them multi-drug resistant TB. If it's a multi-drug resistant TB, then the treatment takes around 18 to 24 months, and through the second-line drugs. And if there is a strain which has resistance to even the second-line drug, the treatment option for extreme drug resistance are extremely limited. I'll glad to tell you that there are 2 new drugs that have been introduced, but they do have their cytotoxicity. Worst is when the person has a comorbid condition and there is a drug-to-drug interaction that can reduce the efficacy of the TB drug as well as the efficacy of the other treatment as well. So the WHO recognizes that we require new drugs to take care of these issues. So with these things, I'm going to very briefly try to tell you that where we have to, what are the molecular mechanisms that are taken up by bacteria for persisting. I'll try to keep it as simple as possible to just give you a sense that how the understanding this molecular mechanism is helping us in designing various kind of intervention strategy. So there are various arsenals, various kind of ways that this Mycobacteria is a very, very clever pathogen that it employs in order to stay inside our body, stay inside of a cell, which is the macrophage is a highly phagocytic, it's a cell, and a milieu of phagosome is so unwelcoming to any pathogen that it is designed to take up the pathogen, chew it completely and throw. Yet Mycobacterium tuberculosis is able to persist inside such a microbicidal environment, and it has several molecular mechanisms attached to it. So I'll talk mainly about a few of them because there is a lack of time before I just try to tell you people about what kind of research we are doing with reference to that. So we'll talk about the blocking of the phagosome lysis pathogen. Mycobacterium tuberculosis resides inside macrophages, is trying to block the fusion between the structure through which it has been engulfed and with the phagosome, which has the enzymes to digest the mycobacteria. So as many of you must be aware, and this is especially for the masters and the research scholars, that the any kind of endosomal compartment, especially when you have phagocytosis takes place, so the endosomal compartment, it fuses with some of the other endosomal compartment which carries a lot of lytic enzymes. So the phagosomes, the early endosomes, they slowly mature into late endosomes, and as they mature into late endosomes, the milieu of the late endosomes become more and more acidic, and they become completely equipped to fuse. When they fuse with the cargo carrying or pathogen carrying the endosomal compartment, they are able to digest them completely. Now, this is what is the general mechanism of when a robust cell is trying to clear the bacteria. But if that is prevented by several molecular mechanisms, then the bacteria can persist inside the phagosome, which and also remain hidden there. So arrested mycobacteria has several proteins that have been identified through very intensive research in several laboratories, that there are specific mycobacterial proteins that can arrest the phagolysosome biogenesis, and the direct cidal mechanism of macrophages are prevented, and thereby help in persistence. So what these mechanisms, now there is a set of proteins that becomes you can target them, you can prevent them from synthesis, you can prevent them from action, and possibly that will help in targeting the persistence of Mycobacterium tuberculosis as well. Yet another mechanism by which MTB can prevent or can persist, and also allows its own dissemination from one site to other, is by controlling the various cell death modalities. We know that the cell death at the cellular level, we call it the programmed cell death as apoptosis. Now, apoptosis, there are several kind of apoptosis nowadays that have been defined, and the apoptosis and the programmed cell death and pyrotosis, which is inflammation-induced apoptosis, are very bad or very detrimental for Mycobacterium tuberculosis. So what it tries to do is it tries to inhibit them. It has several proteins. In Mycobacterium tuberculosis has several active proteins which inhibit both apoptosis as well as pyrotosis. So when it does not allow the cell to undergo death, which is one of the ways the host tries to control, then the mycobacteria can persist better. Necrotosis is on the other hand, is kind of a sudden, more uncontrolled cell death, and it results into dissemination of the membrane of the cell, and that results into that helps in dissemination of mycobacteria. Now, because it helps in dissemination, this is a preferred kind of cell death for mycobacteria, and mycobacteria has certain inducers which can cause necrotosis. And this is what, how intelligent this pathogen is, that it can modulate the cell death modality to its own favor. And if it does so, that the host does not win. Most of the time, as I said, host win, but there can be certain stages at which the host does not win, and mycobacteria can be overwhelming and causes an infection and persistence. Yet another molecular mechanism is to disrupt the balance between Th1 and Th2 kind of cytokines. While talking about the immune response to tuberculosis, I said that it is important that the pro-inflammatory kind of cytokines should be the major response, and that helps in various kind of cell-mediated immunity to take up. So if there is whenever there is a proper Th1 response by an infected person, then there is a MTB clearance. Whereas if there is the response is skewed towards Th2 kind of response, MTB prevail. And there are several factors that mycobacteria releases in order to skew, or in order to divert the immune response instead of Th1 to Th2 response. Now that various labs have recognized such kind of factors, they are working on how to stop such kind of factors from being released, so that the response that the host generates is primarily Th1, and that will clear Mycobacterium tuberculosis. The yet another molecular mechanism that I will touch upon then go into a little bit of our research is the metabolic adaptation. Now, all of you would appreciate that metabolism is a key event which regulates all the physiological processes. We say we are good as good our metabolism is. Professor Anjali Devi also touched upon the various kind of microbiota that are beneficiary microbiota. All of them, gut microbiome, they together through their lot of metabolic changes that they occur, they also have a homeostatic metabolic, they help us in balancing a homeostatic metabolic balance. So metabolism is a very key role in adaptation of this mycobacteria inside the macrophages. So macrophage is the host, mycobacteria. So here it is shown, this is a macrophage. If you all see, these are macrophages. And generally we say that the carbon that is used is, we use glucose, or we use TCA cycle enzymes, and we use TCA cycle. But Mycobacterium tuberculosis, once it goes inside the macrophage, it starts utilizing a lot of fatty acids, and they tend to induce a situation where a lot of lipid droplets start appearing inside the macrophages. And this green color that you see here are the various kind of what you call it as this one is oleate induced lipid droplets. Okay, there are a lot of lipid droplets, and you see that in infected cells, these lipid droplets are much more than in uninfected cells. This red color represents the mycobacterial infected, and you have the blue color representing the nucleus. So there is a completely different metabolism that these mycobacteria starts adapting once they are inside the macrophages. And research show that there are specific metabolic pathways that are important for mycobacteria to survive inside the macrophages. I hope I'm making myself sufficiently simple and clear that metabolic pathway inside the cell, cellular metabolic pathway, are important for mycobacterial adaptation. So I'm talking about mycobacterial metabolic pathway is there are specific pathways that are important for its survival inside the macrophages. And one of the pathway which was shown involves the isocitrate shunt pathway, or which involves isocitrate lyase. And people showed long time, some years ago, that if you remove isocitrate lyase, although it can grow in a culture, if I take an in vitro culture, Mycobacterium tuberculosis, which does not have isocitrate lyase, will still grow. But when it is inside an organism like a mouse model, it is not able to survive. That would mean these pathways are activated only when there is an infection, and they are not really required. They're dispensable when they are growing in vitro condition. So this actually generated a lot of interest in people looking at various metabolic pathway, so much that the new drug bedaquiline, which has been now FDA approved and is being used for treatment of extreme drug resistant TB, actually attacks ATP synthase. And it interferes with the electron transport. It interferes with the ATP synthase activity, and which is still present even in the host. So, with this background, I would like to say that we started recognizing that regulating the metabolism, understanding the metabolism of Mycobacterium tuberculosis inside the host macrophages possibly will give us a number of clues where all we require to target in order to make new drugs. So this is what we started with, and we started looking at how are the metabolic pathways rewired when the Mycobacterium tuberculosis is put into the microbicidal stress of intracellular environment. The microbicidal stress of the intercellular environment definitely includes that there is a nutrient starvation, the phagosomal pH is very low, there is ROS generation, so there is an oxidative stress, and there is also iron deficiency that is created. So we tried to understand how the metabolism changes under these conditions, and we used metabolomics and flux analysis. So when we studied the complete metabolites that are present in these conditions, we cultured the cells, we tried to do the metabolite extraction, then we did metabolic profiling and identified what are the different kind of metabolites that are very specific to one condition. So through various kind of statistical packages, discrimination, we try to discriminate or try to identify that if two conditions here, for example, there's an acidic condition and there is a normal condition of growth, we see that there are distinct metabolites that are present in either acidic condition or oxidative stress or starvation or iron deficiency or condition. And as a result of this, we found that there are metabolic pathways that are perturbed during these specific stresses. Going one step more and we identified around 87 metabolites and we tried to put them into metabolic network. What we found is that an available metabolic network was not sufficient for us to arrive at an inference. So we did a lot of flux analysis as well as simulations were done to see how the pathways are perturbed. I'm going to give you two examples to just give you a sense that what we arrived at. What we arrived at is that there are very when in case of starvation, we found that the TCA cycle enzymes, the mycolic acid synthesis, the fatty acids in the relation, and there are the oxaloacetic acid, which is supposed to be, that is fed into this, comes through from a lot of anaphylactic reactions. But what was very interesting for us to see is that there was an excess of alpha ketoglutarate. And when there is an excess of alpha ketoglutarate, it was seen both in nutrient starvation condition as well as during the acidic condition. But the fate of this increased alpha ketoglutarate was different for when it was, how it was utilized in case of during the acidic condition as well as during the nutrient starvation. Yet another very interesting that we noticed was that it is there is an increased consumption of asparagine. This was simulated, and that asparagine can be converted into aspartate, releasing ammonia, that especially during the acidic media. Now, what can it mean that release of ammonia to Mycobacterium tuberculosis for survival? So what it means is that ammonia can neutralize the acidic milieu of phagosomes. And unlike what people earlier thought, that it may be the urease cycle, which is important for neutralizing the pH, we found that it actually the early adaptation may be through conversion of this excess of use of asparagine and conversion of it to aspartate and release of ammonia. So this particular pathway maybe we should look at, and we should try to find out certain kind of enzymes that we can control or can target. Most of it, the alpha ketoglutarate was utilized through most of this aspartate is utilized via adenylyl succinate synthase, and which is involved in purine and pyrimidine metabolism, which are a very critical metabolism for the pathogen. Similarly, we also found that there is a huge increase in sugar alcohols during the acidic stretch. Now, when we try to extend that, why there should be such an increase in sugar alcohols, what we see is that sugar alcohols, when they are made from their sugar counterparts, they tend to absorb, hydrogen positive hydrogen ions. So that can be yet another way by which they are trying to overcome or adapt to the acidic milieu of the phagosomes. So what we have arrived at, and this is a published data, but we have arrived at a number of very critical enzymes which are important for intracellular survival of mycobacteria. And we are in the process of attenuating the mycobacteria by generating a lot of mutants, so that this attenuated mycobacteria, we will explore their potential as a vaccine candidate. At the same time, because these are enzymes, and if these enzymes are not present in host, can be explored as new drug targets. So for the new drug targets, I think when we have arrived at a very differential distribution of amino acid synthesis during these test conditions, so two of my students were very excited about it, and we started looking at a pathway which is called Shikimate Pathway, which is important for aromatic amino acid biosynthesis and is essential for MTB survival. So we, in that Shikimate pathway, we opted for Shikimate kinase to target and design small molecule. Towards this, we cloned the MTB Shikimate kinase, we purified Shikimate kinase, we designed a coupled assay for it and started screening a number of molecules for that. And although we screened a number of molecules, not very successful in terms of Shikimate kinase inhibitor, but we did some enzyme kinetics to find out there are certain specific molecules that are potential either competitive or non-competitive inhibitors of Shikimate kinase pathway. We further tried to also see whether this kind of molecules, and we came upon one molecule which can regulate the growth of Mycobacterium tuberculosis. So we have now with us a new molecule which has the potential to control the growth of 1.967 to 2.368 micromolar in case of pathogenic mycobacteria. And the good news is that this particular molecule was not toxic to the cell lines that we tested in. So this has given us a very good lead molecule, and we are in the process of exploring this as a potential new target, new molecule or new drug molecule. And this is something which is going on as an ongoing project in the lab. The last one that I would like to tell is about the granuloma formation. As we said, that one of the major questions in the TB is, when a TB is latent, how does it know that immune system is compromised? And then it comes out of the granuloma, it starts disseminating and causes disease. So what we found is that we found a mycobacterial protein, which is the zinc metalloprotein, and this zinc metalloprotein is being explored in the form of a biomarker. But we also see that it's a metalloprotease, and it could dissolve mucin, and at the same time, it is secreted out during the stress condition and helps in migration of cell. So we kind of, hypothesized that if a protein from mycobacteria comes out, it dissolves the matrix between two cells, and it allows the cells, to open up. Does it has anything to do with, dissemination? So we used a zebrafish model and Mycobacterium marinum as a model system to show that whenever this mycobacterial protein, zinc metalloprotease 1, is used, it helps that this increasing, though the injection was done in the head region, it helps in the dissemination of the mycobacteria from head to the tail region. So the model that we are working on is that possibly mycobacteria, when it is inhaled inside, it dissolves the mucin, which can restrict the movement of mycobacteria, And then it allows it to enter. And when it gets and it enters there, ZMP may be, when it is contained in the granuloma, at some point of time when Mycobacteria senses that there is a breach in the immune system, it can it multiply inside the granuloma. ZMP can be released. It may be dissolving the matrix between the cells and help the mycobacteria in disseminating and causing the disease. So this is the hypothesis, this is the model that we are working on and exploring, both ZMP as a biomarker and also trying to see that if we can regulate it. With this, I think I will just summarize that what I was trying to say is that TB is a very, very community problem for us, for India, where it carries a huge burden. And with the development of drug resistance, there's a very requirement of understanding the molecular basis of pathogenesis of this particular organisms, where the understanding their persistence and doing a lot of research there helps us in finding new drug targets, designing new antimicrobials, new vaccine candidate, new biomarkers to have a very early detection of latent cases where the TB is hidden, and definitely add a lot of science to microbacteriology. So it is especially for the student and the research scholar, there are several questions that are unanswered and which requires to be answered so that we are prepared for this particular for this particular pathogen. So we still require to answer several questions like, how does it work with the host system in granuloma formation? Can we identify latency and reactivation? We still don't understand why it gets reactivated. Is there an overlap between hypoxia and gene regulation? And this is one of the best systems to understand mycobacteria host interaction and host pathogen interaction, and how a pathogen responds to environmental cues. These are some of the research questions that are open, and I think the research community should come forward to address them. Now, to conclude my talk, I thought it would be very useful for all of us to just see the, how similar it is that how we are trying to manage SARS-CoV-2 infection. That same thing can be used or learned for TB disease management. If you recognize, both of them are preventive in nature. So if you can have a strong immune system, you do not, you keep yourself away, and maintain hygiene, this can be preventive. Both are curable in nature. TB has a proper therapy, and if it is detected early in time and it is properly treated, complete treatment is taken, then TB is curable. So is SARS-CoV-2. More important, and a message that I would give to all is, whether it is stopping TB or whether we are trying to control SARS-CoV-2 infection spread in the community, is a very collective responsibility. It is important that we recognize and we isolate. We try to make sure that we are not spreading in the disease in the community, and it is not a good idea that it has a stigma where you can attach. You don't try to tell that there is a disease condition. It is important to come forward, tell the disease condition, you isolate yourself, take the therapy, and becomes a collective community responsibility, because as a community, we have to come forward to help these patients. Especially in SARS-CoV-2 kind of pandemic, it is important that healthcare system be available to TB patients who undergo a very long-term therapy and requires a direct observation therapy that is required. The state of immune system is critical for both, and that is something which we require to maintain all the time. Early detection is helpful, so it is important that you be watchful for both the symptoms of SARS-CoV-2 as well as, you isolate yourself. Persistent cough, do not ignore it, because there has been a guideline now given that if a person is having a very long-term cough, even in the SARS-CoV-2 background, it's a good idea to get a chest X-ray done and get yourself tested for tuberculosis. And treatment compliance is very important. Listen to your doctor, complete your treatment. And the most important thing, especially in case of viral infection, please do not pop up antibiotics, because viruses cannot be controlled by antibiotic. If we keep taking pills without any consultation, we kind of encourage development of drug resistance in the community, not only with respect to these pathogens, but also to many other pathogens that are there in our community. So with this, I thank you for patiently listening to me and allowing me to share my thoughts. I end with acknowledging all my clinicians and population study and that we have done, our collaborators and our financial support. Thank you so much. In all possibility, if I had, the physical audience, we keep interacting with each other, but this was almost like a monologue that went on. But that is the new norm. We live with it, and I will be very happy to answer your questions. Thank you.
Thank you, ma'am, for the wonderful session. We thoroughly enjoyed your session. Thank you very much. We will quickly take questions from the audience, ma'am, if they have any.
Kindly stop sharing.
Good afternoon, ma'am.
Good afternoon.
When in your presentation, you said Mycobacterium tuberculosis primarily utilizes fatty acids rather than carbohydrates.
Correct, yes.
So is there any requirement by the organism, because it requires mycolic acids for its cell wall, that it is depending on fatty acids, or any other specific issue that because carbohydrates are very easy to metabolize than fatty acids?
So I think we know that the fats are generally the reservoirs. So the host is trying to deprive the mycobacteria from carbon sources, and can therefore any kind of other carbon sources may get utilized very fast, but they are the reservoirs. And if what mycobacteria does it deregulates the fatty acid metabolism there, resulting in droplet accumulation, and then it can uses that as a source of carbon. Okay, so that's an adaptation that has evolved through over ages when there has been a co-evolution between host and pathogen. So I think there is a survival advantage that mycobacteria gets, because the carbon that is utilized eventually goes through the pathway to synthesize mycolic acid for sure.
Okay. Another question, ma'am. Like in a healthy individual, even pathogen can be at latent stage, right? But
yeah, we call them asymptomatic. It's very difficult to define healthy these days. We say,
but how?
Yeah,
but how these in a healthy individual means immune responses will be very proper, right? But
yes.
But how immune system is allowing it to form granulomas?
Okay, so granuloma state comes only after you have developed the disease, and the load was sufficiently high, and then the body successfully overcomes the disease and then keeps it contained inside the granulomatous structure in the lung, and which is visible. Okay, which is visible when you do an X-ray and you get that this person had a history of a TB disease, not a TB infection alone. And because the immune system with the host pathogen in it, okay, we say that it has been a robust immune response by this particular person who has tried to overcome the disease. Latency comes is that there will be very few cells in which mycobacteria would be surviving, and they may not be even visible in the form of granuloma. Okay, so it can be even some other niches it is surviving, and it is a very slow growing bacteria. It will just not do anything. It will just remain quiescent metabolically. It will be metabolically so quiescent, but the basic things of it to make it survive, it will remain. Okay, and the moment that is a switch which we don't understand even now. What does it sense that the immune system is weak? That it will start slightly becoming metabolic active, and then it's slightly becoming from 1 to 2 to 2 to 3, and suddenly it develops into the disease manifestation takes place. Infection is there. Even suppose I possibly, if I do, possibly I'm infected, but I am not having a disease, so I'm completely asymptomatic, and my immune system is very robust in not allowing the TB bacteria to propagate and manifest itself as in the form of a disease.
Last question from my side, ma'am. Is there any specific reason for selecting zebrafish for your study?
So there are several models that can be used. The reason I use zebrafish is that we wanted to test this at the organism level. When we found that the recombinant zinc metalloprotease 1 that we were studying is capable of influencing cell migration in an in vitro condition in a dish, but we wanted to see that if it happens at the organism level or not. And one of the simple model, and it's a very good model for studying Mycobacterium tuberculosis, using Mycobacterium marinum as a surrogate, because zebrafish, what you call it, when embryo are translucent and the zebrafish itself is translucent, so it is very easy to monitor such kind of events like migration, dissemination through microscopy that is available to us. But eventually, and that is, so it's one grade above the cell culture level that we studied at an organism level, that it happens or not, but it's a very well-established model system for studying at the organism level. Definitely you have a mouse model, you have guinea pig models, and you have higher primates model, which should be, which can be studied. But yeah, I generally restrict myself to up to the mouse model, but here we use zebrafish, which is a very good model at the organism level.
Thank you. Yeah.
Thank you, ma'am. Thank you for answering your questions very patiently. Thank you.
My pleasure.
Ma'am, I'm very grateful about your lecture, ma'am. Please receive, I request you receive our appreciation certificate. Please, ma'am.
Thank you so much. Thank you so much. This is just amazing. It's an experience.
Thank you so much, ma'am.
Thank you.
To present the vote of thanks, I request Dr. Madhusudan Reddy, sir, Director IQAC and Coordinator of the conference, to please present the vote of thanks to Professor Sharmista Banerjee.
Thank you, Ms. Mercy.
Over to you, sir.
Yeah. Professor Sharmista Banerjee, Professor, Department of Biochemistry, School of Life Sciences, Hyderabad Central University. So, ma'am, your talk on the molecular pathogenesis and of Mycobacterium tuberculosis, the highly contagious TB-causing bacteria. So, really, it's very heartening to know that almost 1.5 million are died during 2020, which is next to COVID-19 globally. And much more heartening to know that in India we have got actually 40% of reported cases. Really, it's a very thing of concern where we have to concentrate on this thing. And your lecture, which started with basics of Mycobacterium tuberculosis, and you have shared your really very worthy work, ma'am. So where you have talked about different types of tuberculosis and multi-drug resistant tuberculosis, extremely drug resistant, because we know that TB is a multi-drug resistant variety which is bothering us, but extremely drug resistant tuberculosis and total drug resistant tuberculosis, which is of more concern. Yeah. Mm-hmm. And your research work, where actually you said the role of effector T cells, pro-inflammatory cytokines, and memory T cells, and the role of tumor necrosis factor, interferon gamma during latent infection. Really, it's mind-blowing, man. And you have shared your research where new drugs like bedaquiline, linezolid, and others, and new targets for drug synthesis, which is very important. So really, your lecture is very informative, ma'am, and we are very thankful for you for spending your time. Thank you. Thanks a lot.
Thank you so much.
Thank you, sir. I hope all the attendees have thoroughly enjoyed the session. Let's move on to the second speaker of the day, Dr. Gokul Shankar Sebasan, Faculty of Medicine, at Manipal University College, Malaysia. To introduce the speaker, I would like to invite Dr. N. Kishore Sir, HOD Department of Microbiology and co-convener of the conference, to please introduce the speaker. Over to you, sir.
Good morning once again. I'm happy to introduce our next speaker of this session, Professor Gokul Shankar. Dr. Gokul is a professor of microbiology, Faculty of Medicine at Manipal University, Malaysia. He had extensive career of more than 14 years in research and distinctive expertise of more than 17 years as faculty in microbiology of postgraduates and undergraduates students in India and Malaysia. He started his career as lecturer in microbiology in different colleges as assistant microbiologist and microbiology executive in several industries, lecturer, senior lecturer, associate professor, senior associate professor, and now professor at Manipal University College, Malaysia. Dr. Gokul bagged several awards like Young Scientist Award for outstanding contribution of medicinal plant research at University of Colombo, Sri Lanka, SAB Award of Excellence in Food Safety, Alumni Achievers Award, Award of Excellence in Food Microbiology, Best Teacher Award from MSI India, Pearl Foundation, Best Scientist Award in Microbiology, etc. He had vast industrial experience with several industries like Taj Coromandel, Omlina Bioproducts, New Scientist Aquaculture, Akshaya Foundation, Food Products, Britannia Industries, Relish Snack Foods, Kumar Organic Products, Miso Private Limited, Chola Hill Private Limited, extending his expertise as consultancy services in development of several products and methods. He developed several products by clinical trials like food supplements, herbal health supplements, herbal mouthwash, prickly heat powders, anti-dandruff creams, hair oils, hair creams, anti-acne solutions, fairness creams, deodorant powders, sprays, and many other products. Academically, Dr. Gokul is actively engaged in teaching microbiology. He served as examiner for both the UG and PG students. He is an examiner and paper setter for these students. He is also a PhD examiner. To his credit, he published almost 52 national and international research papers and books. He completed guiding 3 PhD students, and presently 4 students are working for their PhD. He is a member of several professional bodies like AMI India, Association of Food Scientists and Technologists, Society of Indian Human and Animal Mycologists, Blue Cross Society, MSI India, etc. Also, he is a reviewer for several international journals. He is a recognized PhD supervisor of several universities in India and abroad. He organized several symposia and conferences at national and international level. Dr. Gokul also received grants from FRGC Grant Malaysia. With this small introduction, I now take the privilege of inviting Professor Gokul Shankar to deliver his lecture. Thank you very much. Over to Gokul Shankar.
Thank you, sir. Can I share my screen now?
Yeah, please.
Is my screen visible?
Yes, sir. Please
Yeah.
continue, sir.
Okay,
Yes.
thank you. Okay, warm greetings to all of you, to all the delegates, both faculty members and students who have joined this two-day international conference on microbial pathogenesis and recent advances in diagnosis of Omicron. Okay, thank you to Dr. Kishore for a nice introduction. Thank you, but he said a lot about me, not much. I'm a very simple and humble person. Okay, today actually, like you'll be hearing about COVID-19 and Omicron, okay, in detail tomorrow. So I'm not going to talk much about the virus as such, but I'm going to address something which is a little important. Okay, giving you a overview of something which is a collateral damage. Okay, like the main infection, COVID-19. So we just going to be a curtain riser about the fungal infections which are normally there and which has increased during this COVID-19 period. So thank you very much for the senior management of Palamaru University. It's amazing to be there with you all today. And thank you very much to the organizing committee, especially Dr. Shanti Priya, who was in touch with me from the time she planned this conference. Yeah, thank you very much. So with that, let's go on to the topic as such. I hope all of you remember this ship. Anyone wants to identify what ship is this? If you want to unmute and say, or by chat.
Yes, sir, Titanic.
Okay, wonderful. So yeah, all of us know Titanic. And if I ask you the next simple question, like how many Oscar awards did Titanic win? Any guesses? 1, 2, 3, 4, 5. Any guesses? 9. Yeah, that was almost close. But Titanic won almost 11 Oscars. Okay, actually it was nominated for 14 categories and won 11 Oscars. So why did I show this Titanic? And you know what happened to this Titanic? You know, like it hit with an iceberg and then it sank. Okay, so that's the history. But so what's the specialty of an iceberg? Okay, if you have this is for questions for the students who are attending there. What's the specialty for the iceberg? Now, when I ask this question to my students at class, I get all various type of answers, including like iceberg is made of ice. That's also fine.
Titanic.
Yeah, Titanic. Yeah, Titanic sank because of the iceberg, because it hit an iceberg. My question is like, what is the specialty of an iceberg? Okay. So okay,
You can
fine.
damage the ship.
Yeah, anyone wants to try? Okay, fine. Now, by seeing this picture, anyone wants to say? What's the specialty of an iceberg?
More in size.
More beneath the sea.
Although, see, this is, I think, Dr. Sebasan, I completely understand how it feels when you're not able to interact. I'm sorry, I couldn't help interrupting. Thoroughly enjoying your question. I think the tip of the iceberg is what we call it always.
Fantastic,
So
fantastic.
whatever, I should not have, but yeah, I think we all miss this very, interaction which you generally are used to.
Yeah. Thank you, ma'am. Thank you, Dr. Banerjee, for your thoughts. That's what that's what we are missing in these webinars. Fine, I just wanted to keep you all in the same page. That's fine, and thank you for the explanation. So what you see is actually the tip of the iceberg. So it's always good, like when we have an interactive session, like. So fine, what I'm showing you is not the tip of the iceberg that hit Titanic, but it's a iceberg that what we are going to talk today is the medical mycology iceberg. Okay, so what I mean to see is like usually what you see is just the tip of it and the major chunk remains below. So this is very much applicable to the field of medical mycology, one of the neglected fields in microbiology. Okay, I'm basically a mycologist, so please excuse other bacteriologists and virologists. Okay, so there is a famous thing like in the saying which says like in the era of immunosuppression and immunocompromisation, if you do not address the fungal infections properly, then there is a chance that they can emerge as the #1 infections, even surpassing the bacterial and viral infections. So with that, this is the medical mycology iceberg which I am talking about. Okay, so we have been the mycologists all over the world have been putting this issue which is coming up from time to time, and we were seeing like there's an alarming rise of fungal infections, and sorry, why this is not moving. Okay, so also think mycology. It's not that you should not think bacteriology, virology, but also think mycology. Okay, and then that's where the tip of the iceberg, which Madam pointed out just now. Okay, so stop neglecting fungi. That's one lesson which we also learned during this COVID pandemic. Like we should stop neglecting fungal infections. So that was a major thing. Mucormycosis, the black fungus, maiming COVID patients in India. But before talking about, we are also going to talk about the mucormycosis, but we will talk general about the fungal infections in general, what is the prevalence, and then what has happened during the pandemic also we'll address. Okay, so what I'm trying to say is like about the opportunistic infections. So when you say somebody is an opportunist, what you mean is like it's a practice of taking advantage of the circumstances. Okay, that's an opportunism. So these opportunistic mycotic infections are caused by fungi, which are there in the normal air, which we breathe every day, which is there in the environment. These are the fungi whose spores are present abundantly in the environment. The fungi like Aspergillus, Rhizopus, Mucor, and so on. But we do not get infected because our immune system always clears them. But when a person is immunosuppressed or compromised, then there arises a problem. So these fungal infections could be a problem when there is a compromisation in immune system. So in COVID-19, there was also a compromisation of the immune system. So that's what I was trying to tell you, is that these are caused by these infections. Opportunistic infections are caused by fungi, which are of low virulence. Means like normally they do not infect. They are normally saprophytes which live in the environment. Okay, living saprophytes meaning what? Living in dead and decaying organic matter. But provided an opportunity, they cause infection. So I'm basically a Marvel fan, so I just want to show this picture. You know, like Thanos went on to collect all the Infinity Stones, but he chose a time when all these people were not there. Okay, so I don't know, like I would have asked you to identify, but that's fine. So this is Odin, who's father of Thor, and then this is the Ancient One, supposed to be the mentor of Doctor Strange, and you have Ego, the father of Star-Lord. Okay, all these three people were capable of stopping Thanos from collecting the stone. So he chose a time when all these people were not there. So that's how, when a person's immune system is compromised, the fungi takes advantage of the situation, and then it operates. Okay, so why this is important is like, see, if it's in a normal host, these are just numbers. Okay, not very specific. Like if it's a systemic pathogens, you have 25 different species causing infections. If it's subcutaneous, it's 10 species. Cutaneous is 33 species. Okay, but if it's an immunocompromised person, then almost 300 species of fungi can cause infection. And this number is not just 300, like it can keep increasing. Means like any fungi which is there in the environment in an immunosuppressed or immunocompromised person can take advantage and cause infection. So basically, where are these opportunistic fungi? They are. I told you already. They are saprophytic in the environment. They live in the dead and decaying organic matter. They are also endogenous in the sense they can be a part of the commensal flora. Okay, but can turn pathogenic in circumstances where the immune system is compromised. So the question of coming, like when you say like what are these opportunistic fungi? Like I would say, like you can name any fungi starting from A to Z. Okay, I told you I'm already a Marvel fan, so I just put from Ant-Man to Zuras. Okay, they are Marvel characters. Something like from A to Z, Aspergillus to Zygomyces, anything can cause an infection. So doesn't mean that for each alphabet I have a fungus there. Like there will be a fungus, but don't ask me that for each alphabet. Can you tell me a fungus name? Right. So normally, opportunistic mycosis. We see an upward trend in the opportunistic mycosis, which is attributed to increased clinical awareness, improved clinical diagnostic tests, also because of invasive diagnostic tests. Okay, that's what we do these days. Like say you have a you suspect an ulcer. Those days we don't we go in for treatment straight away, but now the person is put on an endoscope to check whether there's an ulcer actually. So we go into more invasive diagnostic and therapeutic procedures. Okay, that's also a reason for the upward trend in opportunistic mycotic infections. So, but the most predominant reason being an increase in the susceptible hosts. So the number of susceptible hosts has gone up because of the COVID infection. Okay, so all these are in general the reasons for a weakened immune system. It can be immunosuppression or immunocompromisation. So the varied reasons can be from infections like HIV, or it can be a debilitating disease like cancer or diabetes, or a person is in chemotherapy, radiotherapy, and all that. Okay, but COVID-19 is recently added to the list. Okay, so before we go into the impact of COVID-19 and the fungal infections, let's see what's the most common fungal infection. So you all know that beauty is skin deep, or is it deeper than the skin? Yeah, but this beautiful skin can be the appearance of the beautiful skin can be spoiled. Okay, the inner beauty cannot be spoiled, but can be spoiled by the most common fungal infections. Okay, they are the superficial mycotic agents most of the times, the dermatophytes and the other agents. The most common one being the dermatophytes. You also know that dandruff is a fungal infection, okay, which can also be a major cosmetic concern. Right. So addressing on these dermatophytes, these are fungi which infect the skin. Basically, they live on keratin, so they are keratinophilic. So you know that the layers which are rich in keratin in your body is the skin, the nails, and the hair. So these dermatophytes can colonize on these layers and cause these infections, which are called the ringworm infections. So basically, the name ringworm or tinea is actually a misnomer. Okay, ringworms are not caused by worms. Okay, and tinea again, like you hearing the name tinea, like people may think it's a parasitic infection. So, but that's actually a misnomer. Like, but because of the popularity of the term, like it's still being used in the medical circles and textbooks even now. Okay, but it's a fungal infection. So, as far as the geographical distribution of the dermatophytosis, it's worldwide. It doesn't spare any culture, any race, any country. Okay, so it's fair with everybody. So it causes infection almost everywhere. But in India, okay, we use this word called the Great Indian Fungal Epidemic. Means like the epidemic with the dermatophytosis is never over. Okay, it keeps on, and then it's like our great sagas like Ramayana and Mahabharata. No? Why this great is because it's prolonged for a long time. Okay, the infections, you keep seeing them again and again. The one reason being India is India's climatic conditions, a hot and humid climate. Okay, which is also the condition of most of the Asian countries like Malaysia. Okay, that's one predominant reason which contributes for the fungal infections. Okay, so you get these infections also by from your pets. Okay, so we always have, when we have a pet, a dog or a cat, okay, we pet them and we cuddle them, we sleep with them in the bed. Okay, but make sure that they don't have the fungal infections. Or they are going to be the transmitters of fungal infections to you. So these transmission of fungal infections from the pets, especially the pet dogs and cats, is very common, okay, as far as dermatophytosis is concerned. Right. So what are the nature of these dermatophytic infections? I just want to show you a few pictures for you to understand, because there is a popular saying that every person in the world will get at least almost 90 to 95%. Okay, so every person will get at least one form of dermatophytic infection in their lifetime, whether they realize it or they don't. Okay, so it's very important that all of you should be aware of these dermatophytic infections. And they are scaly pruritic infections causing itchy nature. Okay, and as I told you, like, they are common in hot and humid climates. Okay, based on where these infections occur, it's classified differently as Tinea captis, corporis, gruris, and also we will see some pictures of that, so you'll understand that much better. Okay, this is Tinea corporis, where the infection is on the glabrous skin, on the smooth skin of the body. Okay, and then it does not involve the hair. Anywhere in the body you get these infections. Okay, the ringworms rings because of the typical nature of the circular regions we get because of these dermatophyte infections. That's why they are called as ringworm infections also. This is Tinea pedis. Okay, this is one of the common infections. Okay, and then it's also called as athlete's foot. And it's not just attributed only to athletes anymore. Like earlier, I told you, like hot and humid climate like is favorable to dermatophyte infection. When you use athletes, most commonly they wear shoes, tight-fitting shoes always. So that's the reason, like, because of the sweating, they get the infection. But these days, everyone wears shoes. It's not the athletes alone. So, but popularly known as the athlete's foot. Okay, so we usually say, no, you never judge a person like unless you step into their shoes. Like that's a famous saying. But make sure that if you want to step into their shape, make sure that they don't have a athlete's foot or a Tinea pedis infection. So what I mean to convey here is, like, the infection can be acquired by fomite transmission, by sharing of things like sharing of linen, sharing of bedding, okay, cloths, towels. Okay, so dermatophyte infection can be transmitted. Okay, so this is Tinea pedis, and then you will see this more prominent in the intertrigeminal region here. Okay, and that's because I told you a lot of sweating. Okay, basically you should understand, like wearing of shoes, okay, is not suitable for a climate such as India, Malaysia, and all that. Not most of the Asian countries. Okay, like it was suitable for countries like UK, US, and all, because it's a cold weather, so they need to protect themselves. So they had but later it has become a part of our dress code that we all need to wear shoes. Okay, and finally we end up getting these infections. Right. So this is Tinea unguium, like the infections of the nails. Okay, and then there is no need for any nail polish. Like all your nails get colored. Okay, with the fungal infections, and this is one of the most terrible form of dermatophytes infection. The sense, like, it's very difficult to treat because the active fungus goes and sits deep in the nail bed, so it becomes very difficult to treat the Tinea unguium infections. Okay, normally most of the dermatophyte infections, what you see on the skin, can be treated with a topical application of antifungal cream. But whereas the nail infections, as I told you, they are deep-seated, will be difficult to manage. Okay, that's what just now I told you. Okay, so this is one case of intractable case of dermatophytosis, like, and that too in an immunocompetent women. That's one case, like, that was during the COVID time. Okay, so there you can see these lesions here. Okay, and this is Tinea corporis. Okay, the ringworm lesions, and then the typical appearance of these lesions is like you will have the raised margins on the surface. Okay, so we say it's a well-defined margin. Okay, that's the way the clinicians, I mean the dermatologists, identify these infection. So and then obesity, okay, even and other conditions like wearing tight-fitting clothes and all can contribute to the dermatophyte lesions. Okay, this is tinea captis, where the infection is on the scalp. Okay, so if like a lesion is extensive, sometimes it's called tinea favus, where a person may lose the hair also. Okay, so tinea cruris. Okay, this is also called as joxis. That's in your private parts. Okay, see, this is some infection, like people are very embarrassed to discuss with others. Even sometimes they get this infection, they do not go to the doctor. Like, what you need to understand is, like, it's a fungal infection, like how you treat your bacterial infection or a viral infection. Like, you're not well, you have to go to a doctor. Similarly, because of the occlusion, mostly and a lot of sweating, you get these infections very commonly in the private parts. So there is no need for any embarrassment. You get these infections, they are basically fungal infections. Then what you need is meet a dermatologist, okay, or a skin specialist, okay, to get the appropriate antifungal treatment. Okay, so that's again pictures of Jock Itch. Okay, so you will see a lot of guys putting both the hands inside, inside the inserting both the hands inside the trousers. Okay, so usually it's popularly we make it a sort of a humor, saying like whether the person is actually stylish or it's because of the Jock Itch, because the itching is so very common, like you cannot resist. Right. So now this is something which is appearing now because of the COVID infection, which is called the mask tinea. Okay, actually it's tinea faciae. The infections tinea infections which you get it on the face is called tinea faciae, which is very, very rare condition because you don't get much of sweating on the face. Okay, that's a very rare manifestation when compared to the other forms of tinea. But this is becoming more common because of the wearing of mask constantly. So that's the name mask tinea. Okay, so this is not much of studies has been published because the importance is given for more life-threatening infections. Okay, these are novel infections which attack only the skin surface. So not much of published data, but I understand. Like I was talking to one of my dermatologist friend in Chennai. He was saying like he's getting a lot of fungal patients with fungal infections, especially dermatophytosis after the COVID era. So this is a picture which I showed you just now. That's a study from the Milan city, where for 10 years almost, they had just very few patients reported, almost 5 patients in 10 years reported with tinea faciae. Okay, I told you that's a very rare manifestation, but in a very short period, they saw, like in October 2020 to 2021, okay, the number of cases are more. So, and then when these patients were interviewed, like they found that these patients were wearing these masks 6 hours a day minimum. Okay, and most of these people were wearing the same mask up to 10 days. So please watch out. Okay, it's not necessary, it's okay alone to wear a mask, but make sure that if it's a disposable mask, it has to be disposed. Okay, don't use the same mask for a very long time. Okay, and then if it is a washable mask, okay, and then you have to wash it regularly. Right. So this is again another case of like where you say, like I was talking about tinea cruris in the private parts, tinea corporis in the body, but this is something which is a wide spreading infection. And then this is something which is very common now, which is a terbinafine resistant form of trichophyton. Trichophyton is a dermatophyte which causes these infections. So you can see that almost all over the body, the person has got this dermatophyte lesion. Okay, so this is something which is a new danger because of the terbinafine resistance. Okay, so the common precautions, like what we do for these infections is like we say it's a rule of 2, where you are supposed to, if it's going to be a fungal infection on the body surface, what you do is like you apply a topical antifungal cream 2 centimeters beyond the margin of the cream. I told you actively growing fungus will be on the margin. Okay, so 2 centimeters beyond the margin of the infection, you need to apply your cream. Do it for twice a day for 2 weeks. So that's the rule of the 2. Okay, and then like we all believe in the modern dresses like jeans, jeggings, and all that. Okay, but all those are not suitable for us. Okay, wear loose cotton garments. That's what suits the climate of India. And that's besides other things, like there are other precautions. Let's not go into that. Okay, so this is something, an interesting study report from Rutgers University. Okay, where they say like more than half of the people who visit a beauty salon, okay, they come back with a skin infection or a fungal disease. Okay, so which means like next time you go to a beauty parlor, let's check out. We believe in natural beauty, right? So I mean, Indians believe in natural beauty. The next time you go out for a beauty parlor, okay, make sure that you don't end up with a fungal infection, right? Okay, so that's one thing, like one of the most common infections. So then I want to highlight a few of the other opportunistic fungal infections that can manifest in the post-COVID era. Cryptococcus Neoformance is one organism, okay, which is it's an opportunistic fungal organism, like it's a yeast, okay, which is common. It's common, has its reservoir status, okay, in all the birds, especially the pigeon drops. Okay, and pigeon drops, you see it in most of these places, like along with the residential area, like when you go to the old temples, monumental buildings, churches, everywhere you see these pigeon droppings. And and every time, like we have done some studies on the pigeon droppings and the prevalence of Cryptococcus Neoformance. Like we found a lot of I mean droppings which we isolated, like it had Cryptococcus Neoformance, but never mind, like most of us are immunocompetent, they are not going to cause us any harm. But in an era where we are living with possibility of immunocompromisation with COVID-19, then you need to watch out this fungus. Okay, so this is a study which was published way back in 2004, where we first demonstrated that these fungi are present, okay, not only on pigeon droppings. That was reported quite frequently. Like we found that it's in other a lot of other birds, like including the most common bird, crow. Okay, so that was the first report that we reported, that the crow droppings had Cryptococcus Neoformance. Okay, so this is one of our one of the recent studies published in 2022, just now. Okay, so they said, like similar to our previous study, they said even the domestic birds, a lot of domestic birds, which means like the pet birds which you can keep at home, okay, they can harbor they can be the source of Cryptococcus organisms, which can be a potential cause of Cryptococcus. Why this is a problem? Like because Cryptococcus, though it's an opportunistic fungal pathogen, but when it infects an immunosuppressed person, okay, it can cause a life-threatening condition such as cryptococcal meningitis. It can affect the meninges of the brain. Okay, so that's some of our paper reports. It says bird droppings can be fatal for patients. Okay, and okay, something which I always tell about Cryptococcus Neoformance. This is something funny which I tell about Cryptococcus to my students. So I thought I will share with you also. Like we see this sort of structure. This is a negative staining of Cryptococcus Neoformance. So in negative staining, what you stain is the background, not the cells as such. So this is with India ink staining. You see the cells of Cryptococcus Neoformance. I always say, like, whenever what do you see on a full moon day? Questions for you. What do you see on a full moon day? Fantastic. You see a full moon on a full moon day. Okay, so whenever you see a full moon next time, okay, remember it, try to correlate with Cryptococcus neoformance. So what you see is this moon, okay, that is a yeast cell, and what you see as a halo zone around the moon, okay, on a full moon day, okay, that is actually the capsule of the fungus. So this is a capsulated yeast. That's one virulence mechanism this organism has got, which can escape the macrophages digestion. Okay, that's why they are able to cause debilitating infections like pneumonia and meningitis. Okay, so the comparison is just like I always tell my students, like whenever you see a full moon, okay, one full moon on a black sky, then you are seeing a full moon on a full moon day. But if you see multiple full moons on a black sky, then probably you are looking into a negative staining of Cryptococcus neoformance. Okay, so now these are some recent reports, like in 2021 and 22, where it says like the Cryptococcus neoformance. This is what I was highlighting. Like, this is a pathogen, which is an opportunistic pathogen. It was causing earlier infections in HIV people, but now after the COVID-19 infection, so like COVID-19 is also a sort of a predisposing factor for this Cryptococcus neoformance. So yeah, this is again cryptococcemia in a patient with COVID-19. So it can manifest again. So that's something which you need to remember, that this is another fungus, which is because of the environmental distribution. It's commonly present in the environment, so it can develop into a life-threatening pathogen. Okay, so there are already reports, like, then we need to watch them more cautiously. Okay, so we were discussing about two types of organisms. One is the organisms which live as saprophytes in the environment, and then they enter into the human system and cause infection. Okay, that's first group. Second group is like a group which is a normal flora, okay, which is endogenous in the body, okay, but can transfer themselves from a normal flora to a pathogenic flora, okay, provided there is an opportunity of immunosuppression and immunocompromisation. So normally, like systemic candidiasis doesn't happen in normal person, but candidiasis is quite common, an infection. Like you see it in infancy, you see it during pregnancy. Okay, those are the common conditions where you see the candidiasis. Vulvovaginal candidiasis is very common during pregnancy. But whenever a person has been predisposed, okay, with conditions like prolonged steroid therapy, prolonged antibiotic therapy, okay, where there are therapies, and even diabetes mellitus, okay, so those things can predispose a person from a normal candidal infection to systemic candidiasis. Same thing happens today with a lot of cases of candidemia, okay, and systemic candidiasis being reported, okay, in the post-COVID era. Okay, so that's a picture of Candida for you to see. Okay, and then now usually we say these predisposing factors are the post-operative immunosuppression, IV catheters, okay, cytotoxic use of cytotoxic drugs and all that. But now COVID-19 is also added to the list. Okay, so it's sort of a vicious cycle. Like normally we see, like, whenever there is an infection, you take a lot of antibiotics. Okay, these antibiotics are going to wipe out your friendly bacteria. Okay, that's going to give up the place or a chance or an opportunity for the other organisms, okay, to multiply. Okay, that's what happens, okay, in vulvovaginal candidiasis, where the wiping of lactobacilli, the friendly bacteria in the vaginal tract, okay, promotes the growth of Candida, causing vulvovaginal candidiasis. Okay, so like oral candidiasis, the oral thrush can be used even as a marker for HIV, because that's a marker for immunosuppression. Okay, we will not be surprised in future that if people are going to use this sort of oral thrush or oral candidiasis as a marker for other sorts of immunosuppression, may be applicable to COVID-19. Okay, which we need to wait and see. Okay, so this is what I was telling you about the oral thrush. Okay, you get the creamy white plaque, curd-like plaque on your tongue. That's oral thrush. Okay, don't get confused with the normal whitening of the tongue, which is because of the unclean tongue. Okay, and this oral thrush can be used as a marker for HIV. So but remember, I told you, don't get confused with unclean tongue. Half a knowledge is always a dangerous thing. So if you tell this, I always tell my students, next time don't ask somebody to show their tongue and say you have got a white tongue, then go and get yourself tested for HIV. Okay, so you need to know the difference. Okay, but this is a white creamy curd-like plug. Okay, which you get it on the tongue. Okay, so the common agents of candidal infections are the most predominant ones being the albicans tropicalis glabrata cruzi. It's now renamed again. And the most prominent one, which is gaining importance in the current world, is Candida auris. Okay, also put as a superbug. Okay, usually you know that the word superbug is used only for bacteria. We always call the drug-resistant bacteria as superbugs. Okay, you don't use the word normally as superbug for a fungus. Okay, but this is the first time, okay, for a particular fungus like Candida auris, the word superbug is used. Let's see why. Okay, so Candida auris is one emerging global threat. Okay, and why? Because I told you, it's a multiple drug resistant organism. Okay, some strains are resistant to all the available form of antifungals. There are 3 major classes of antifungals. So all those antifungals, it's resistant. Okay, and then it is most of the times it is misidentified because there's no proper protocols to differentiate them. But there are molecular techniques being developed now. Otherwise, you may be required to send it to a standard laboratory for identification. If it's in US, CDC is giving free service to submit the Candida auris infection. But I don't think we don't have much of the facility here in countries like India, where you have a reference laboratory to go back. But a lot of research is going on still, to differentiate Candida auris. Okay, because most of the times in culture, okay, they can be misidentified as other members of Candida. Okay, the other major problem with Candida auris is usually in a healthcare setting, they can spread very easily. So most of these outbreaks, okay, in the healthcare settings were usually, up until now, it was because of different drug-resistant bacteria. But now we have included this fungus also into the list. So that's one other major concern, okay, in the healthcare settings. Then why you need to worry is the normal disinfectants may not be effective in eliminating it. Some of the normal disinfectants may not be effective in eliminating. Okay, and normally this Candida auris can be carried on the skin surface. So that is one dangerous thing, but this does not happen to most of the other organisms. So especially the fungal organisms, this can be carried on the skin surface, so it can be spread to others. And other thing is the most worrisome part is usually Candida auris before the COVID time was a common problem. Okay, only when in a long-term care facility where there is a lot of hospitalization, or in elderly people's treatment and all that. That's a place Candida auris was reported. But now you see Candida auris be getting reported more commonly in acute care also. Means after a trauma, a person gets admitted, okay, and undergoes an operation. Okay, even there you get reports of Candida auris. So that's something to worry about. Okay, so this is a fungal report. Okay, why Candida auris is so important is this is a report about a patient, okay, who was admitted in the Mount Sinai Hospital for almost 90 days. He received treatment and died. Okay, so that's not important. What is important after that is they were trying to disinfect this room after the patient has died. They were trying to clean. They had to recur a lot of special cleaning equipments to clean this room. Okay, why? It was there, the Candida auris, the remains of Candida auris was there almost everywhere in the room, right from the mattresses, the bed rails, the door, the curtains, the sink, the whiteboard, the phone. Okay, everything in the room was infected. Everything was having a positive, testing positive for Candida auris. So they had to finally go into a level that they have to rip out some of the ceiling and the floor tiles to eradicate the fungus, completely eliminate this fungus from that particular room where the patient was admitted. Such is the tenacious nature of this organism, Candida auris. Okay, that's where the concern is. Okay, so just for fans of Pushpa, okay, so don't think it's just Candida auris. If you call Candida auris, it's just sounds like soft like a flower, but it's actually a fire. Okay. Right, now this is a CDC data where it says Candida auris reports. This is from February 15, 2021. Okay, where all that marked area is full of reports from Candida auris. Okay, but now they stopped updating this data. It's not available after February 15th, 2021. You know why? Because it has become so common and widespread that there is no need to update the map because it's globally present everywhere. Okay, so the antibiotic resistance, based on antibiotic resistant threats. Okay, usually I told you, like whenever we talk about antibiotic resistant threats, okay, or the antimicrobial resistant threats, we always think about bacteria, okay, not about fungi. Okay, but among the bacteria, okay, so they have also put in as the major threats, next to the carbapenem resistant Acinetobacter, which is the first. Okay, in the US, they think Candida auris, okay, which is resistant to multiple antifungals, will be a major threat, and that's an urgent threat as well. Again, this is the data from CDC, where it says like the isolates from countries like India. See that? That's something worrisome. Okay, it's resistant to all this. Fluconazole, that's the most common one being used. Amphotericin B, okay, resistance. Okay, fluconazole resistance is almost 93%. Okay, echinocandins, at least there is a comfort that the resistance is not complete. Okay, so that's one of the choice of drugs which can be used for treatment of Candida auris. Okay, so echinocandin will be the first line of treatment, and then there are a lot of other things which are being tried now. Okay. Okay, this one is a new drug, Ibrexafungerp. Okay, actually it was introduced in June 2021. Okay, approved by the US FDA. Okay, so that's one of the drugs which is being explored for treatment of Candida auris. Okay, so besides this Candida auris, okay, so why other? There are a lot of other fungal infections which also hamper the COVID treatment, is because a lot of symptoms of the fungal infections, okay, especially the fungi which can cause pneumonia like Aspergillus, okay, the symptoms are very, very similar to that ones of the COVID-19, which is the fever, cough, shortness of breath, and all that. No, so that's very common. So it will be very difficult, and then laboratory testing will be necessary to find okay, whether a COVID patient has got a co-infection with fungi, with an Aspergillus. Aspergillus, I told you, it's a fungi which is ubiquitously present in the environment. So a person can inhale the spore and get the infection. Very easy, as simple as that. But we may be actually addressing the treatment of COVID-19 where we will be ignoring that the person may also have a co-infection with Aspergillus. So that's something which will be a challenge, okay, in future, because the invasive nature of Aspergillus infection or invasive candidiasis, which are the most common fungal infections which we encounter in COVID-19 patients. Okay, so the other thing is like these fungi, I told you, they are available freely in the soil, so they can be acquired by breathing in. So all those systemic fungi like Histoplasma, Blastomyces, okay, Coccidiomyces, okay, all those fungi actually they live in the soil. Okay, they are all dimorphic fungi. Dimorphic, you know, right? They can exist in two forms, like a mold form and a yeast form. Okay, when they invade a human host, and how they you acquire them is just by inhaling the spore. So it's easy. It's there in the environment. So you inhale the spore and you get the infection. Okay. So whenever the immune system is compromised, then the infection is more severe. Okay, and be life-threatening. Okay, so that's it. And then about mucormycosis, like I think most of you would have followed a lot of articles and write-ups about this black fungus in the media. Okay, where the black fungus actually the media is always crazy about naming organisms, you know, like calling them black fungus, white fungus, green fungus. No, fungus appears in all colors. Okay, and as I told you, like any of the opportunistic fungi can cause a infection in a immunocompromised person. So you give any color, there'll be a fungus. Okay, so it's going to be all colors, different colors, multi-dimensional. So even WebGR like fungus and all, it's going to be reported later. Like so this black fungus, okay, though it's a misnomer, like you call all these fungi which are dematiaceous fungi which produce the dark pigments. Okay, they are called dematiaceous fungi. You actually call them black fungus, but now it's a popular term, so it's okay. So mucormycosis is caused by the members of mucorales. Mucorales consists of mucor, rhizopus. All these fungi are where you find them. They are in the environment. Okay, they normally grow on the bread. Okay, so it's a bread-eating fungus. Now it's become a flesh-eating fungus. Okay, so they can cause debilitating diseases. Okay, and then most of these diseases, like the mortality can be up to 50%, like if it's not properly managed. Okay, the manifestation is like starting from a normal sinus infection, then it can end up in a loss of vision. A person can lose eyesight because of mucormycosis, or a person can lose his whole life. Okay, and then there's a lot of debate of why this mucormycosis suddenly emerged during COVID-19 and all. It's actually, this is not going to be a surprise for all the mycologists, because from time and again, mycologists have been telling that when there is going to be immunosuppression and compromisation, there is going to be emergence of all these opportunistic infections. So this time it was mucormycosis, so it can be anything else. It can be aspergillosis or any other fungus, okay, or something like the Candida auris in future. So we need to watch this space. This is one of the articles mine, like where that was last year, like when it was the mucormycosis was famously talked about. Right, and then other less known fungi, which is earlier called Penicillium marnifi, it's called Telleromyces marnifi, which causes Telleromycosis. Okay, this is another pathogen which is an emerging pathogen, especially in the Asian countries. Okay, also in India. Okay, it's again a dimorphic fungi. It looks like this. Okay, it produces a cherry red pigment. Okay, on the medium. Okay, so very colorful. Right, and so dimorphism. Whenever I talked about dimorphism, I always show my students this picture of Hulk. Okay, you know Hulk is a dimorphic. What are the two different forms of Hulk? Two different forms of Hulk. Yeah, Hulk can be normally as a Professor Hulk, Professor Banner. Okay, and then he can be an aggressive form called the Hulk. Okay, but similarly, this fungus, the dimorphic fungi, Telleromyces, can be in a mold form and a yeast form. Okay, what makes the Hulk, I mean Professor Banner, to become a Hulk is the anger. So whenever he is angry, he becomes Hulk. Okay. Same way, if you change the temperature from 25 degrees to 37 degrees, okay, this is thermal dimorphism. Then the fungus, which is in the mold form, converts it into the yeast form. Okay, why this is important is because the human body temperature is 37 degrees. So that is an adaptation by these fungi to cause human infections. Okay, so this is again a picture of the Saccharomyces magnifi. Okay, so and then Aspergillus is other organism. Okay, I think exceeded time, so we will stop here. Like that's Aspergillus is another major infection. Okay, which will be very in the post-COVID era. Also because of the use of a lot of pesticides, aerosol-derived pesticides and fungicides in the environment. Okay. We'll stop there. So mycologists have more fungi and more fun in the era of immunosuppression and immunocompromisation because a lot of fungi to study. Okay, with that I conclude, and thank you to the organizers for the opportunity.
Thank you, sir. That was an amazing talk. All of us
Stop sharing.
must be careful regarding this masthenia which you have introduced to us. Thank you very much. Let's quickly take a Q&A session, sir.
Yeah, sure.
We received one question from Shrav in the chat box. Is there any other reason other than immunodeficiency to get fungal infection after COVID? That was a question posed by Shravani.
Okay, the prolonged use of steroids. Okay, that was one of the reasons that was believed to be for the uprise of mucormycosis in India. Okay, because the early start of steroids during the initial stages of COVID infection, not now, because those times the physicians were also struggling to find like what sort of treatment is appropriate for this infection. So those were days like the steroids were used inappropriately. That's one reason for the upcoming of mucormycosis. Okay, other normal conditions can be even diseases like diabetes. Okay, where a person can be susceptible to fungal infection.
Okay, sir. Thank you.
Yes. Hello.
Yes, sir. Carry on, sir.
Good afternoon, sir. Sir,
Hi, sir.
when we see this black fungus, that is mucormycosis, such cases were suddenly increased more during the second phase, not during the first phase or recent Omicron strain cases, sir. So
Correct,
what is the reason, sir? Is that because in the second phase they have used more corticosteroids so that immune suppression was done and this fungi infection has been increased, or any other reason, sir?
Yeah, actually there were a lot of factors that would have contributed. One is the prolonged use of steroids or inappropriate use of steroids, and then the other factors could be the uncontrolled levels of sugar. Most of the times it was correlated with diabetes because the more focus was addressing the virus as such. Okay, so they were not very eagerly looking into the other parameters, which is into the holistic health. So it was coinciding with much with diabetic people and steroid treatment. That's two major reasons. And then there are a lot of other hypotheses saying like because of the use of industrial oxygen and all that it erupted. But I don't agree with that because mucormycosis during the COVID time was not reported only from India, though the numbers were high. It was even reported in US. Almost 6 to 7 states in US reported of mucormycosis during. There is an uprise of mucormycosis in after so many years in America. So there are the major reason is the immunosuppression and compromisation. Okay, and also like they also believe that the susceptibility. Okay, because it's an inhalation infection. A mucor infection is an inhalation infection. The susceptibility was more because of the damage to the respiratory tissues by the COVID virus as well. So that could also be a reason.
Thank you.
So we received another question from Dr. Vasavi ma'am in the chat box. Does prolonged usage of fluconazole lead to resistance? That was her question.
Yeah, actually, like there are two ways, like we say, like the azole resistance is two ways. One is an environmental way of resistance where you use a lot of azole drugs in the environment as on the plants basically. Okay, and then the fungi which is existing in the environment, they get resistant. That's one way, which is more common. The other way is like if a person is in a prolonged treatment, then there's also a chance that the organism can gain resistance. So there are two ways of resistance. So yeah, prolonged usage is one of the ways here.
Sir,
May I have a quick question?
I'm sorry. Yeah. A little different question from written this COVID-19 and all, sir. So you might have heard about this one, so dieback disease in neem, which is a fungal infection.
Yeah.
So why suddenly it started affecting only neem? Because we consider neem as a most antimicrobial producing substance, whether you take a leaf or bark or whatever you take, nimbin, nimidin, all the antimicrobial substances which is produced by neem, but still it is affected by this fungi, and that too only neem trees, not other trees. Is there any specificity, sir?
I'm not sure about I work mostly with human pathogenic fungi. I'm not sure about the plant pathogens, but see, it is like we call so many things as antimicrobial. That's fine. You call turmeric as antimicrobial, but you know, like fungus can grow on turmeric. Okay, so the antimicrobial effect is not 100% always. Okay, see, when you say something is an antimicrobial, it can be an antimicrobial for a virus, it can be for a bacteria. It is not something which is universal, which will be for all the microorganisms. So that way, it doesn't work there.
May I ask?
Yes, ma'am. Yes, ma'am.
Yeah, I think you touched upon a very, very important area which most of us in the society has ignored. I think fungal infections are not talked about much. I wanted to just further ask, are there zoonotic fungi which can come from the animals? Because many a times we are ignorant about, still go eating and not recognizing that something is infected with fungus and consuming it, and can it percolate into the society, into the community through animals, like we know about bacterial and
yes, ma'am. Actually, for zoonotic infections, like when I mentioned about dermatophytosis, I said like it can be from the pet dogs and cats, like what we get the dermatophyte infection. But most of these fungal infections, when we talk about the systemic infections, like the route of entry for these infections are by the respiratory means. So means like if I have aspergillosis, okay, I cannot transmit aspergillosis to another person in the ward, to the doctor or a person who is attending me. Okay, unless if it's a viral infection or mycobacterium, if the people who are with them, okay, can get infected because of aerosol. But for fungal infection, the most interesting aspect is like when you want to acquire an infection, you have to inhale the spore from the environment. Okay, so it's
apart from inhalation, suppose consumption, suppose there is a fungal infected or not really fungal, but something which is contaminated when we eat something, can it also?
Yes. A lot of aflatoxins are there, like the aspergillus derived toxins in the foods and all that. There are a lot of regulations, like when we have the import of pulses and cereals, for the presence of aflatoxins, because
sometimes there is a white, those kind of. So that should be
correct. But the interesting aspect is like most of these fungal infections which infect plants do not infect human beings. That's a good news also.
So I'm now talking about mainly animal. I mean, sometimes we consume, but I do not know how much people are aware or how much of the work is going on in this aspect, because I feel that we don't know that when we get in, whether maybe they infect us, they are cleared from human system, but then can they also become opportunistic at some point of time? So
Correct. I agree, ma'am. Like, because all like the normal flora of the human beings, like the normal flora of the animals, also consists of a lot of other fungi. So there is always a chance that these organisms can have a chance to encounter a human host. Only difference is, like, as far as the immune system is strong enough, most of these
I think most of them are
pathogens,
opportunistic, and that is one good thing, that possibly immune system should be able to take care of it.
Yeah, only when there is an immunosuppression or compromisation, then there's a problem. Otherwise, it's fine. Yeah.
Thanks a ton. Enjoyed listening to you.
Thank you, ma'am. Thank you very much for your comments.
Hello. Ma'am, there is one question from Rajshekhar sir to Shamista ma'am. Ma'am, please, if you allow us.
Yes, please. Yeah.
One question.
Good afternoon, ma'am. Madam, with respect to the tuberculosis and in context of India, and in 1950s, the total population of the disease people about 250,000. At that time, as per my knowledge, and go through some literature, I come across that at the time our PM Nehru was called a scientist from London. J.R. Bignall is one of the famous pulmonologists at the time. And he came to the India and he given a suggestion that rather than go for the medication, give a good nutritional food to the those selected or diseased people, and it will be eradicated within 5 years. But if you look at the 1950 to 2022, and we have a strain which has a highly resistant almost all drugs, and the tuberculosis is a opportunistic disease when with reference to the AIDS patients also. And if you look at the total population of AIDS, that is 35 million across the globe. But India alone consists of near to 50 million children which are suffering from the nutritional immunodeficiency syndrome. So rather than treating the diseases, and if you go with the some other alternative that provide a good nutritional food to the people, so we may eradicate the TB from India. And can you comment on this?
Yes, sure. See, while you are making a very good point that malnutrition or compromised immune system is one of the reason why TB infection gets manifested into disease. We can see what you are talking about is a prevention. Okay, let us talk that if you continue to take lead a very healthy lifestyle, try to avoid take a proper nutritious food, have a state of mind which is healthy and a healthy body, you do your exercises, this takes care of the homeostasis of the body as such and prevents many infection, which also includes TB. Okay, even if you get infected, I will not say prevent infection. Let us say even if you inhale TB, you will not have the disease because your immune system is strong. Now that cannot be a consistent thing. While you can always supplement, there can be situations when you are in some or the other form have become little bit compromised. Your body has become a little bit compromised. It may not be only because of malnutrition. Okay, it can be also because of several other things. Let us say I'm allergic to spores. Okay, and in certain season I am allergic to spores. That's my physiology. And in some or the other form, my health is not as good. Okay, and that's a compromised situation for me. So therefore, when we are talking about eradication of a disease like tuberculosis, which spreads through aerosol, I think SARS-CoV-2 has given us enough experience that even for those patients, possibly it would have really worked if instead of having a stigma attached, that's a person is a TB patient, we also wear mask, we also isolate ourselves, we eat healthy food, we maintain hygiene, but at the same time take treatment. Treatment is taken because the load of the bacteria is very high. So body alone cannot fight. So you have to give a treatment so the bacteria is killed and then body clears it for a small duration of time. So that the bacterial load, the moment it comes down, body once again starts recovering and comes back to homeostasis. So we can prevent it by keeping our immune system strong. To eradicate, there will be always, if you remember my slides, I have been always saying only 10% people have a disease, let us say. So 90% people are cured. They have able to contain their infection because of their immune system. So that's a very good news. That's why people say that apart from treatment, give supplements, give immunomodulators. We call them it's a huge research. Immunomodulators of plant origin, Ayurveda, there's a big, huge research going on there that can we have certain substances which will stimulate our immune system, keep it healthy, have a modulator, and that will be not only for one disease. Good thing is it may not be for one disease because you are supporting the system, so it may be able to take care of many such infections that come us in. And then the treatment can be infection specific. But the immunomodulators and supplements can be still for general. So yes, you made a good point that prevention is possible and always prevention is better. But unfortunately, under certain conditions, if somebody has met with an accident and is in the hospital, also is a compromised person, under those conditions, we still require a very robust, treatment. The way community can help with treatment of any pathogen is do not take antibiotic over the counter and eat. This is one thing which we all can do. We all can spread this message. What happens is, I have a flu, which is a viral infection, but I think I'll figure out, take some antibiotic because I'll feel better. It does nothing, but it may just help the microbes which is inside me, opportunity to get selected, the microbe which is resistant to it. Other microbes will die, but the resistant microbes, so what have I done actually by taking that pill is that out of 100, if 2 were the resistant, I have killed the 80, but 2 are the one which are going to multiply. And now I have a population of drug resistant one. Okay. So this is where I think the education has to go. And if you start a medication of a course of 5 days, do not interrupt it in the middle, because the concentration of antibiotic requires to build up in the body in order to have effective concentration for killing the bacteria. So these kind of practices actually helps in selection of drug-resistant strain, which is actually a man-made creation situation. These are the things which we can avoid, and I think through education and if somebody is aware, should spread these things. So treatment and prevention require to go hand in hand, and very careful usage of medication as and when required with proper consultation should be done. That will take care of a lot of infections, disease, which are even emerging new. Drug resistance is a major problem.
Yeah, very true, Professor said, Mr. Banerjee, ma'am, because recently, even during this Omicron menace actually, so there is, in social media, everyone, they have forwarded, the list of drugs which are need to be taken for COVID infection. So even just for common cold also, they have started using azithromycin.
That's what I'm saying. That how does it help? People do not just know, and these are the kind of things. If as an educated set of people, if we can just spread, then I think that is something which we all can do. And that is where I was saying that communicable diseases is also a community responsibility. That's why it's called communicable. Okay. So I think it's a responsibility. If we want to control, we cannot say this policy will do, that policy will do, and no, it doesn't work like that. And it is not only for SARS-CoV-2, which has made us aware of these things, but I think this should have been followed for a long, long time. And if you recall, our parents have been telling, please, haath dho ke khaiye. This is nothing new that we know. But so it's something which we have been brought up with, but we tend to forget. And I think these are few things which consciously, if we do, we will be able to control these kind of spread to a larger extent. Throwing things, let's suppose sometime if you have a set of antibiotic and I have thrown it in a water body, that also helps in selection of resistant strain in that water body. Now that can come in some other form inside human or in animals or in plants. So it's a very, it's a cycle which we can avoid, which is a man-created, human-created problem. So antibiotics are popped in even in the poultry, in things for various reasons. And these are the places where we require to be, create an awareness that if we very rampant use of antibiotic can lead to a huge drug-resistant problem, and eventually, even if it is not immediately affecting us, will affect human beings at certain. It will come back to us in some form or the other, because bacteria exchange their DNA left and right. They will exchange the resistance from environment bacteria to a pathogenic bacteria. We will not know, but it will come back. And then we say new strains are coming, and then we say new emerging infections are coming. At some place, possibly we have been responsible for it, and should be very, very careful about it. It goes for any pathogen, whether it's virus, bacteria, fungal, or anything. Parasitic problems, even that, it comes. So I think this is something which all have to remember. Yeah, don't follow that list of huge lists that comes. Body takes care of it. Good thing is that SARS-CoV-2 is largely being taken care by body, so that is good.
Thank you, ma'am. Thank you.
Thank you, ma'am. One more question to you from the chat box.
Please.
Does calcification in lungs get completely removed after getting cured from TB?
Although I don't think we can say that it can completely get cured, because completely is, I think it just leaves an impression. And that helps the clinicians to know that if there had been a previous history, because some part of the lung tissue gets damaged permanently, and that scar shows. Because that is not then you cannot regenerate lung, so that scar shows. And yeah, I think it doesn't get completely. The tissue doesn't heal up. That's one problem with the lung infections, and if there's a damage, then that part of the lung does not recover. If there are clinicians around, maybe they can add more.
Thank you, ma'am. Thank you very much. Thank you, sir, for taking up a QA session. So please accept our digital appreciation certificate. Thank you very much, sir.
Thank you. Thank you.
Next, to present the vote of thanks, I would like to invite Dr. Shantipriya Ajmera ma'am, the convener of the conference, to please present the vote of thanks. Over to you, ma'am.
Dear Dr. Gokul, I am very happy.
Madam, unmute yourself.
Mute. Dr. Gokul, I am very happy about your wonderful and informative presentation. Gokul, he was one of my good friend. In your presentation, you touched about the importance of addressing fungal infections, which are airborne and opportunistic, arised because of immunosuppression during COVID, and about opportunistics outnumber normal pathogens, various reasons for immunosuppression, along with the geographical distribution of dermatophytes, and types of fungal infections and the transmission, and treatment methods for infections due to for long hours wearing a mask, importance of wearing loose and cotton clothes, and debilitating infections which is caused by the Cryptococcus Neoformance and Candida, which is a multi-drug resistant and global threat. And also about the antifungal drugs, that is new drug, Ibrixifungal SCY78. And also invasive nature of fungi, which is life-threatening like mucormycosis. And explained very well about that characteristics of that dimorphism in changing their morphological based on the temperatures and adaptation of fungi to cause human infections. We are very glad about your presentation, sir, and very happy. I'm very thankful to you. Thank you so much.
Thank you. Thank you for the opportunity.
Thank you, sir. So I thought in future we will plan offline. That's better to interact each other, to spend more time to discuss.
Sure.
Yes, yes, sir. Thank you so much, sir.